Hello, my dear friends. Welcome to Live Prep. Myself, Dr. Gajapati Verma. As I informed you earlier, under the name of Bootcamp, we are providing both revision lectures and also we are conducting test series. Revision lectures are available both on Live Prep app and also on Live Prep YouTube channel. But the test series can be given only through the app. And now with this, now let us quickly go with revision of a new chapter, that is breathing and exchange of gases. So in the human physiology, they have mentioned about this. So this process with respect to human we are studying now. And in the beginning of this particular unit, they have given some information, and majority of the names they have used here already we have discussed, and the information is not that much complicated. So this is simply for understanding purpose they have given. So simply read and understand. So here I am not going for explanation of this particular part. And now we are going with the chapter, that is breathing and exchange of gases. So here the thing is, so what is this breathing? Breathing is also called as a respiration. What exactly we are doing in the respiration? The thing is, atmospheric air is taken inside the lungs and there exchange of gases will occur and then some air will be released outside. So that's why here under this external respiration process, so we are talking about inspiration. Atmospheric air is taken in, drawn in into the lungs. So that's why it is called as inspiration. Now CO2 rich air is released outside into the atmosphere, that is called as expiration. So generally we are focusing on this, but not only this, we are also talking about exchange of gases. How these exchange of this oxygen and CO2 are taking place at the level of alveoli and at the level of tissues. These two things also we are discussing. And along with this, we are also talking about transport of gases like oxygen and CO2 also we are studying. Clear? But the last part, that is called as cellular respiration, that separately we are studying under a separate chapter, that is called respiration in higher plants. And please remember that respiration concept, even though you are studying under plants, the same concepts are applicable even for animals like human also. So please remember this. Anyhow, in the NCERT also at one location they have mentioned about all these points. There also once again I will tell you. But the main question here is why we need to take this atmospheric air. And in that atmospheric air, what is our major focus? What is the gas that we are taking inside and what is the major gas that we are releasing outside? So these are some important points, and for that in the NCERT they have given some basic introduction. See, so even though in the new syllabus, so they have deleted digestion and absorption chapter, so there is a small relation here. Now let us try to understand this. We are taking food, and this food is ultimately used as energy source. This is what the ultimate conclusion is. The food that we are eating is ultimately used for energy production. Yes. And what is that energy form? That is the ATP energy. And from these food materials like carbohydrates, fats, proteins, how this ATP is produced. And this information you will study under respiration chapter. Yes or no? And there you are studying about glycolysis, Krebs cycle you are studying, and also you are talking about electron transport chain or electron transport system you are studying. Clear? So here generally for our understanding purpose, so we are generally starting with the glucose molecule here. Not only this, even the proteins and fatty acids are also, fats are also related with these cycles, and ultimately they are also useful for energy production. But here we are starting with the glucose. Let us quickly go through this. So glucose is ultimately converted into pyruvic acid in some sequence of reactions, and this complete pathway is called as glycolysis pathway. Clear? And in this glycolysis pathway we are talking about NADH production. Clear? Remember this. Next one is regarding the Krebs cycle. Under aerobic conditions, this pyruvic acid is converted into acetyl coenzyme A, and now this is going into this Krebs cycle. In the Krebs cycle also you are talking about production of NADH, and along with this you are also talking about FADH2 production. These are some molecules that are produced, and now these NADH and FADH2, NADH and FADH2. So these here succinate dehydrogenase is there, no? Here FADH2 will come, and these are entering into electron transport system or electron transport chain, and ultimately they are involving in ATP production, and this is what the overall relation is. So in this way food materials are ultimately involved or used for production of energy, that is ATP. Clear, up to this it is a different concept. So how we are taking the food, how it will be digested, how it will be absorbed into our body, all these will be discussed in digestion and absorption, but that chapter is deleted, it's okay. But once it is taken inside the body, now how it is used for ATP production, that concept you are studying in under separate chapter called as respiration. You need to understand this interlink. Now here, why we are talking about breathing here? What is the role of breathing here? Now you need to understand this. And this is the process, right? And this is the process. And this in this process called as electron transport system. So here if you are clearly observing, here at this complex number 4, you can understand that half O2 plus 2 H plus ions will along with this, along with all these things, now ultimately it is involving in production of water molecule. Clear? And here I don't want to talk about these internal details because separately we will study under respiration. But you need to understand that under aerobic conditions, this oxygen is required here. Please remember here, whatever the oxygen that we are taking inside our body, where exactly it is used? It is exactly used in electron transport system at the complex number 4 for the formation of water. If water is produced like this, it is called as a metabolic water. Okay, anyhow, under respiration chapter we will discuss about this. This is called as metabolic water, and you can see this reaction, and this is the reaction exactly where this oxygen is utilized, and for this reaction we need oxygen. This is what the ultimate use is. So, and from where we can get this oxygen? From the atmosphere. So that's why we are taking atmospheric air into our body. Understood or not? This is one thing. And in this way our body is utilizing the oxygen. Nice. And where exactly the CO2 is produced? Observe this. This is the Krebs cycle. In this Krebs cycle, if you are observing, when this alpha ketoglutarate acid or alpha ketoglutarate, anything you can say, when it is converted into succinic acid in this reaction, 5 carbon is converted into 4 carbon compound. And what about that remaining carbon? It is released in the form of CO2. Please observe this carefully. So in this way, in some of the reactions, this CO2 is released by our body. Our body means our tissues. Our tissues means cells. So cells are ultimately they are utilizing oxygen and they are releasing CO2. This is what the ultimate conclusion that you need to understand before we are starting with this particular chapter. So let me quickly tell you this. So in our cells, so at some reactions oxygen is utilized. That's why we need to take oxygen from the atmosphere. And in some reactions CO2 is released, and this CO2 is harmful for our body. So that's why it should be released from the body. So that's why now during expiration CO2 is released. And this is what the ultimate thing that you need to understand. So for these things, how oxygen is entering into our body, how it is undergoing exchange at alveolar membrane and exchange at tissues, and how it is transported in the blood, all these things we need to study in this particular chapter. These reactions we are not studying in this chapter. So remaining all the things we are studying in this particular chapter. I hope now you understood what is the relation between these reactions and then why we are doing this breathing. Clear? So in this way some basic information they have mentioned in NCERT. And then they are saying that, so in different organisms, so different mechanisms are there for the exchange of gases. So they have given some NCERT examples, at least NCERT examples you need to remember. In the case of sponges, coelentrates and flatworms. So in these organisms, specialized structures are not present. So that's why this exchange of gases is directly happening by simple diffusion across their body surface. Directly across the body surface, by the diffusion process, these exchange of gases are taking place. Next one, in the case of earthworms, so moist cuticle is helping in exchange of these particular gases. Next one, in the case of insects, a separate network of tubes are present and it is called as a tracheal tubes system is present. So this information you can link up with even cockroach also. So this is very important. So please remember this. So this type of separate system is present. And please understand, in the case of tracheal tubes system, what happens is atmospheric air is directly going to the each and every tissue. In the case of insects like cockroach, at least cockroach information is important for us. So here what happens, atmospheric air is taken in and it is directly distributed to each and every tissue. So exchange of gases is taking place directly at the tissues for insects in the case of tracheal tube system. But in the case of human, it is not like this. Atmospheric air is first coming into lungs only. From the lungs it should go into the blood. Through the blood it is distributed. So that's why in the case of these insects like cockroach, even the blood is technically called as hemolymph, and that is not playing any role in oxygen transport in the case of insects. But in the case of human, yes, blood is very important and it is playing important role in this exchange of gases or transport of gases. Understood or not? This is one important point that you need to remember. Anyhow, when we study separately about the cockroach, there I will tell you separately. Next one, aquatic arthropods and mollusks. Then these are coming under the invertebrates, and in the case of vertebrates, the fishes and all these things. So they are using the gills for exchange of gases. If gills are used, it is called as a branchial respiration. So please remember this. Next one, in the case of terrestrial forms like amphibians, reptiles, birds, means aves, and then mammals. In all these cases, it is the lungs. Lungs are present. They are involving in this particular breathing process. So, and if lungs are involved, it is called as pulmonary respiration. Okay. If skin is used, it is called cutaneous respiration. If lungs are used, it is called pulmonary respiration. If gills are used, it is called as bronchial respiration. If the network of tubes are present, it is called as tracheal tubes type of respiration. So these are some names that they have mentioned in NCERT. Along with this, under the example of amphibian, they have separately talking about the frog. Frog information related to frog is also important for examination. So please remember this. And in the case of frog, since it is amphibian, anyhow, pulmonary respiration through the lungs it is taking place. This is compulsory. But in the case of frog, specifically if you are talking along with the lungs, here skin is also involving, moist skin is also involving in exchange of gases. If skin is involving in respiration, it is called cutaneous respiration. And along with this, buccopharyngeal respiration is also taking place. So it is the buccal means mouth, and it is leading to the pharynx, right? At that location, the epithelium is also involving in exchange of gases. That's why it is called a buccopharyngeal respiration. So that's why in the case of frog, all these three types of respiration we are observing. Clear? So this is what another important information that we need to understand. So with this, some basic introduction is completed, and now we are focusing on human respiratory system. So what are the various components that are present within the human respiratory system? So in the case of human respiratory system, so it is starting with the external openings called as nostrils, which are present above the lips. These nostrils are present. So these nostrils will open into the nasal passage, and this nasal passage will open into the nasal chamber, and this nasal chamber will open into the pharynx. And already we studied about this one in detail in the regular classes, right? So pharynx, it is the common opening for both mouth and also for the nose, and both of them are having the common opening that is called as pharynx. So the part that is towards the nose is called as nasopharynx, the part that is present towards the mouth is called as oropharynx, and then the part that is towards the larynx is called as laryngopharynx. So in this way, total 3 parts we are studying, but this pharynx is the common passage for both mouth and nose. So this is what. And now from this pharynx, now it is from the pharynx again 2 openings are present, yes or no? So let us have some rough diagram. All these are rough diagrams only. So now let us see like this. Suppose this is the mouth, and suppose this is the nose opening, and here they are having some common passage that is called as pharynx. And here at the throat, in general we are calling it as a throat, and here you just see it facing like this. Now what happens? This is the ventral, and behind this will be the dorsal, right? On the ventral side what is present? Now here the trachea will be present, and here on the dorsal side what is present? Esophagus is present. Okay, and now here the opening for this trachea is called as glottis. Opening for this trachea is called as glottis, whereas the opening for this particular esophagus is called as gullet. So please try to understand these names. So now what happens? Now the opening, the pharynx is opening into the trachea through the glottis. At this opening, there is a cartilaginous flap, and that flap is called as epiglottis. Epi means above the glottis. That's why it is called epiglottis. And what is the role of this particular epiglottis? Now here, when we are swallowing some food, maybe liquid or solid, whatever it is, when we are swallowing the food, now this epiglottis will close this glottis so that the food material will not enter into this windpipe, so that it will enter into the food pipe only. So this is what the significant role. So in this way we are talking about this. and this. Now let us continue further. And here at that particular opening, now here we are studying about a structure called as a larynx. It is also called as a sound box. In this sound box, some vocal cords are present, and the vibration of these vocal cords is responsible for making some sounds. Okay, so this is some information that we need to understand. And now this larynx is opening into the trachea, and this trachea will further divide at the level of fifth thoracic vertebrae into primary bronchi, left and right primary bronchi, and this primary bronchi will further divide into secondary bronchi, and they will further divide into tertiary bronchi, and then they will further divide into bronchioles. And in this way, in the bronchioles also, generally we are directly talking about the terminal bronchioles. Divide, ultimately they are divided into terminal bronchioles. But please remember, when we are talking about initial bronchioles, technically we are not writing about initial bronchioles, but in NCRT, separately they are talking about initial bronchioles when they are talking about the C-shaped cartilaginous rings. Okay, there they have used this name. So if you are adding that name here, so what you will get? Trachea will divide into primary bronchi, they will divide into secondary bronchi, they will divide into tertiary bronchi, they will divide into initial bronchioles. Please remember, technically we are not using this, but since in NCERT separately they mentioned it, let us add this. So initial bronchioles, they will divide into terminal bronchioles, and after that you will get the respiratory bronchioles, and then again they will further divide into some small ducts and all, and finally they are opening into some sac-like structures called as alveoli. So this is what about the complete sequence. Clear? And this is what the NCERT diagram that they have mentioned. So forget about this trachea. Let us start with this primary bronchi. Primary bronchi, from there onwards the branches up to the alveoli. All these things on one side is one lung, other side is the other lung. Clear? Whenever we are studying about these human structures, please remember, so since you are the viewer, for you it is right side, it is left side, but whenever we are talking about the human structures, you should not see like this, it is like this. For me it is the right side, for me it is the left side. So this is the right side, this is the left side. For all the human structures we need to study like this only. It is not with respect to the viewer, it is with respect to the one who is teaching you. So in this way, this is the right side, this is the left side. So in this way only we are talking about this. So this is the right lung and this is the left lung. And anyhow, here also you can see between this the heart is present, and though it is present at the center, it is slightly tilted towards the left side, and to accommodate this particular heart there is a particular small notch is present in the left lung. In the left lung there is a small notch to accommodate this particular heart. So that's why overall size, if you see right lung is larger when compared with the left lung. Anyhow, all these points one by one slowly we will understand here. And here also they have given some labeling also, so please understand this. And when we talk about the thoracic cavity, at the time once again I will tell you here. So for the thoracic cavity, when we are talking, what we are saying, on the dorsal side vertebrae are there, ventral side sternum is there, on the lateral sides ribs are present, and on this bottom side, so you can also call it as posterior side, here diaphragm is present, and this diaphragm is a muscular structure. So in this way it is present. Slowly one after the other we will study. So this is about the human respiratory system that they are talking here. Next one we are talking is regarding the C-shaped cartilaginous rings. And in the case of NCERT, what they are saying here, so these C-shaped cartilaginous rings are present, and what is the role of these particular rings? So please remember, when we are doing expiration, exhalation, then we are releasing the air. So you just imagine a tube-like structure. When air is released, then what happens? Now it will try to become flat. It will try to become flat. Please remember, if it is completely closed, during inspiration, air cannot enter into this. So that's why at least some portion should be open. Even though during expiration, even though air is released out, the tube should not be completely closed. That means it should not be collapsed. It should not be collapsed. So some structures are needed for partial opening of this. And what are the structures that are helping in partial opening of this? These C-shaped cartilaginous rings are helping in partial opening of this. If there is a partial opening during inspiration, air can easily enter into this. So this is what the role of this particular C-shaped cartilaginous rings. And where these are present? They are present in trachea, primary bronchi, secondary bronchi, terminal bronchi, tertiary bronchi, and also initial bronchioles. Up to this they have mentioned. In the NCERT they have mentioned like this. So for NEET examination, please follow NCERT sentences. So up to this. That means terminal bronchioles. Initial bronchioles up to this cartilaginous rings are present, but terminal bronchioles and then the remaining that respiratory bronchioles and then alveolar ducts and all these things, there these cartilaginous rings are not present. This is the sentence that you need to interpret from this NCERT sentence. Clear. Next one is regarding the alveoli. So finally from that, after the terminal bronchioles, so alveolar ducts will be present. From that alveolar ducts again some branches are coming out, and finally they are opening into some irregular walled structures, and they are bag like or sac like structures. They are called as alveoli. Singular is alveolus, plural is alveoli. So they are opening into this. So these alveoli, they are again connected with this blood capillaries. So blood is passing through this, so that exchange of gases can occur at this particular alveoli. Clear? So this is what one important point that we need to understand. Next one is regarding the nose in general. Now let us start with one after the other quickly. So regarding the nose, if you are talking, so now these are the nostrils or the external nares. They are opening into nasal passage, and then they will open into the nasal chamber. Whatever the NCERT information is there, up to that only you please remember here. Here also some nasal bone is present, some cartilages are present, but that list is not needed for NEET examination. In higher classes, you will study about these things. Here they are not needed. Clear? Next one. So now in the nasal cavity, if you are observing, now here there are some ridges are present, and these are called as conchae, superior nasal conchae, middle nasal conchae, and inferior nasal conchae, and these are increasing the surface area, so that now here some humidification can take place, and then regulation of the temperature also will take place for the air. So many other uses are also there. We don't need to go into those internal details. And here also, if you are observing here, so here some nerves are also present in this here. Nerves are also present in this particular portion, so that nose is involving in recognition of the smell. So this is what that part. So here these are called as olfactory nerves, and here is the olfactory bulb, and these are related with detection of the smell. This is another function, but actually we need to study about this under a separate chapter called as neural control and coordination. But at our level, we are not talking about this smell mechanism and all, so we are not going into those details. Simply you just remember this information. Now air is passing into this, and here there are some openings into the pharynx. These are called as internal nares. So these nostrils are called as external nares, and here the openings are called as internal nares. Now they are opening into the pharynx. Now in this pharynx again three portions are present. So now the portion of the pharynx present towards the nose is called as nasopharynx. The portion of the pharynx that is present towards the mouth is called oropharynx. The portion that is present towards the larynx is called as laryngopharynx. So in this way pharynx is divided into three parts for our easy understanding purpose. Now from this pharynx, now it will open into the trachea. Now here we are talking about these particular parts. So within the nasal cavity, vestibular region is present, respiratory part is present, and olfactory part is present. So overall if you see within this nose, what we are observing, external nares we are observing, vestibule, that means it is a small cavity that is present, it is called a vestibule, and then nasal passage, nasal chamber, nasal conchae are present, and internal nares are present. Now this internal nares will open into pharynx. This is what pharynx, again three parts, just now we have studied about these three. And now it is opening into this larynx, and larynx is also called as sound box. So within this some vocal cords are present, so vibration of these vocal cords will result in making some sounds. This is some general information. Some of these points were not mentioned in NCERT. Even though you are not referring that, there is no problem at all, but whatever the sentences they gave in NCERT, at least those sentences you should remember. Clear? Next one. So this is also general diagram. So different diagrams I am showing, that's it. And this is the epiglottis. So it is present above the glottis. It will prevent the entry of food into the windpipe, so that food will go only into the food pipe or esophagus. This is what the important information is. Here also different types of cartilages are present, and you don't need that list of cartilages. Please remember that list of cartilages is not there in our syllabus. Unnecessarily some students are getting worried about what are these cartilages, what is their number, yes, and where they are present. So that list is not needed, so simply leave it. Coming to the next one. So now it is opening into the trachea, and this trachea is going almost to the middle of the thoracic cavity. This is the thoracic cavity, right? Middle of the thoracic cavity, and approximately at the level of fifth thoracic vertebrae. At the level of fifth thoracic vertebrae, the trachea will divide into right and left primary bronchi. This is what they are saying, right and left primary bronchi. Sorry, this is what they are saying. So it is divided into right and left primary bronchi, and now they will further divide into secondary bronchi, they will further divide into tertiary bronchi, they will further divide into bronchioles, and finally bronchioles are several times they are divided, and finally they will open into that alveolar ducts, and then finally they will open into some sac-like structures called as alveoli. So in this way, now this is the left lung and this side is the right lung. Clear? This is what the complete branch that we have already discussed, and these are the cartilaginous rings, and this is the windpipe, and this is about the esophagus, this is the food pipe. Clear? So this is on the dorsal side, this is on the ventral side, this is the ventral side, and behind this is the dorsal side. Clear? This is what the diagram, rough diagram. Please understand, you don't need to worry about labeling of each and everything, but whatever the NCERT diagrams they have mentioned, for you need to understand NCERT diagrams and the labeling. That part is important, but for this type of diagrams from the other sources like internet, you don't need to worry about what is the each and every labeling. You don't need to worry about it. Understand the basic concept, try to remember that, and try to interlink with mechanisms. That's it. Clear? Sorry. Next one we are talking is the bronchial. This is what we have discussed till now. Clear? So in this way, finally they are opening into some sac or bag like structures called as alveoli. Now here, these are some diagrams that I have given here for understanding purpose itself. Clear? Next one. So here lobes of the lung, not mentioned in NCERT very clearly, but if you can remember. Left lung is a little bit smaller and it is having only two lobes. Upper one is called as superior lobe and the bottom one is called as inferior lobe. But on the right side lung, these two are also there, upper superior and the bottom inferior are there. Along with this, on the right lung there is an extra lobe that is called as middle lobe or median lobe. So in this way, in the right lung 3 lobes are there, left lung 2 lobes are there. Reason also you can understand, right lung is larger when compared with left lung, because in the left lung there is a small notch that is present to accommodate the heart. That's why this notch is called as cardiac notch. Anyhow, if you can remember or else leave it, no problem. This is some basic information. And finally, what we are saying, for this lung at the end on the superficial surface, now there are two membranes are present, and these membranes are called as a pleural membranes. And these membranes, the outside membrane is called as a parietal pleura and the inner one is called as a visceral pleura. Parietal pleura and visceral pleura. And between these two membranes there is a fluid that is called as a pleural fluid. And what is the function of this pleural fluid? It will reduce the friction between these two layers when the heart is undergoing these volume changes. Clear? So this is what about this particular structure. And this outer one is called parietal pleura, that is closely present with respect to the thoracic layer, and the inner one is called as visceral pleura, that is closely present with the lung. Clear? This is what the arrangement of these layers. Next one. So for this particular respiratory tree, so if you are talking about the conducting part and respiratory part is also called as exchange part. So from where to where this conducting part is there, from where to where this respiratory part is there. Now observe very carefully. So in this tree, what we are starting with the nose, and we are finally ending with the alveoli. Nose means external layers and finally ending with alveoli. Now in this, only the last part means technically what will come here. So in the bronchioles also, last part of bronchioles are called as respiratory bronchioles. So respiratory bronchioles, alveolar ducts and alveoli, only this part will come under respiratory part or exchange part, because exchange of gases will occur only in this particular portion. That means whatever the portion that is before to this, that means until the terminal bronchioles, now the exchange of gases does not occur. Air is present inside these also. Please understand this, air is present inside these also, but the air that is present inside this particular portion is not involving in exchange of gases. So even though not mentioned in NCERT, there is some other concept that is called as a physiological dead space. So though air is present inside this, it is not involving in exchange of gases. So you can consider this as a dead space. Yes or no? So in this way, but the last portion like respiratory bronchioles, alveolar ducts and alveoli, and this portion is involving in exchange of gases. That's why it is called as respiratory part or exchange part. And the part present before to this is called as conducting part. Simply it is bringing the air till this particular place. That's it. That's why it is called as conducting part. So from where to where it is present. So please understand very clearly. At least try to understand these with respect to NCERT names, because in higher classes you will add some other names also. So that's why you may be confused with respect to those names. So that's why at this level at least remember the information with respect to NCERT names. Clear? So this is what, these are the two important names that we need to understand. So in this way, respiratory bronchioles till alveoli, this is the exchange part, and before to this is the conducting part. That's it. Clear? Next one. Next one we are talking about is regarding the thoracic chamber. Why we need to talk about thoracic chamber first? Because immediately after this we need to talk about mechanism of breathing. In the mechanism of breathing, we will talk about changes in the volume of thoracic chamber. So that's why first we need to talk about thoracic chamber. What is the significance of this particular thoracic chamber? Very, very important for examination. Observe carefully. Now this one, from the starting from this particular neck portion until this diaphragm, it is called as thoracic chamber. This upper part is called as thoracic chamber. Now observe very carefully. Now here anyhow for this particular thoracic chamber, upper part at the neck it is completely closed. At the neck it is completely closed. Clear? And now on the posterior, this is anterior side, let us consider. For the thoracic chamber, this is anterior, this is posterior. Clear? And then this is dorsal and this is ventral. Please remember the names also. Antero, posterior, dorso, ventral. In this way only we are using the names. So please try to understand with respect to NCERT names. Now on the anterior side, at the neck portion, it is completely closed, tightly closed. Tightly closed. Some blood vessels and some tubes are passing through this, but the remaining portion is completely closed. It is not open into the thoracic chamber. So on the anterior side it is closed. But what about the posterior side? On the posterior side, what is present here? Diaphragm is present. This is what? Diaphragm is a muscular wall. Diaphragm is a muscular wall. It is made up of muscles. And the muscles of this particular diaphragm are called as phrenic muscles. Muscles of diaphragm are called as phrenic muscles. Suppose this is the thoracic cavity. Now this is what the diaphragm. Diaphragm. Muscles of diaphragm are called as phrenic muscles. Phrenic muscles. Sometimes they may use this word, phrenic muscles of diaphragm. Don't get confused. It is the name of muscles that are present within the diaphragm. Diaphragm means what? Diaphragm is a general term. Diaphragm means it is like a wall. It is like a wall. So that's why it is called diaphragm. But actually it is made up of phrenic muscles. So here on the posterior side, what are present? Phrenic muscles that are forming the diaphragm. But anyhow, on this anterior portion, now it is. So here, please understand here. Now suppose this is the esophagus. Esophagus is passing like this. It is coming into the stomach portion, right? So now it is present like this. It is not opening anywhere here. It is directly going here. Now here it is, suppose the trachea. Now it will divide here, and now it is forming lungs here. Yes or no? In this way they are forming. So please remember here. So there is no opening here. There is no opening here. So on both these sides it is closed. Nice. So anterior posterior side it is closed. We studied. Now we will talk about dorsoventral sides. Now on these sides what happens? Dorsal side vertebral column is present. Ventral side sternum is present. On the lateral sides ribs are present. In this way they are present. If you clearly observe this, vertebrae, now they are many in number. Sternum is present. Between these many ribs are present. Yes or no? But now you need to understand that how this gap is closed. Once again this gap is closed by muscles. Please understand this. So here this gap, suppose here are the vertebrae. On the dorsal side you need to imagine. Okay, on the ventral side here is the sternum. Yes or no? This is how it is present. And how they are connected? Now these are connected like this through the ribs. Through the ribs they are connected like this. Now what we are saying? How the gap between this is filled? Now this gap between these is filled with muscles. Some muscles are present here. What are these muscles? Suppose these are the ribs. These are the ribs. On the outside of this, there are some muscles, they are called as external intercostal muscles. Inter means between, between costal ribs. Okay, so external, on the outside, between the ribs, some muscles are present. They are called external intercostal muscles. These are external intercostal muscles. So inside also there are some muscles. They are called as internal intercostal muscles. So in this way, there are some muscles that are filling this particular gap. So in this way, now on this sides also, dorsal, ventral, lateral, anterior, posterior, all the sides it is closed with the muscles. So since it is completely closed, it is an airtight container. It is airtight container. Please understand this. This point is very, very important for examination. Please understand this. This thoracic chamber is an airtight container. Why we are talking about airtight container here? Now you will understand this particular logic here. The thing is, suppose this is the thoracic cavity. Suppose this is closed here. Now in this, if you are talking about this, suppose here is the heart, heart is present, okay. Regarding the heart, let us first understand. Regarding the heart, let us understand. Please remember, heart is having its own muscles. Heart is having its own muscles, and what are these muscles are called as? Cardiac muscles. Since heart is having its own muscles, it can undergo its own contractions and relaxations. Yes or no? Heart can contract on its own because it is having its own muscles. So there is no problem for the heart, but the problem is for the lungs. For the lungs, there is a problem here. I am drawing some rough diagrams, observe carefully. For the lungs, there is a problem because the lungs, during this exchange of gases process, now what happens? Atmospheric air should come into the lungs. Yes or no? So oxygen rich atmospheric air should come into the lungs, and CO2 rich alveolar air should go into the atmosphere. So in this way, some movement of gases should occur here. For the movement of gases, observe carefully the logic. I am interlinking the logic, so understand this. For the movement of these gases into the lungs, out of the lungs, for these things, lungs also should undergo contraction and relaxation. This is what the basic. When lungs are undergoing contraction and relaxation, it will be easy. But the problem here is, can lungs undergo contraction and relaxation on their own? The question here is, can lungs undergo contraction and relaxation on their own? No, they cannot. But heart can undergo its own contractions and relaxations because it is having its own muscles. But in the case of lungs, for the contraction and relaxation of the lungs, there are no muscles. This is what the problem is. So since lungs do not have muscles for contraction and relaxation, now what is happening? Now these lungs, they are depending on the thoracic chamber. They are depending on the thoracic chamber. So now what is the meaning here? This thoracic chamber is a closed container. Closed container. Now let us see the principles of chemistry here. So in the principles of chemistry, what we are saying, for a closed container, if the volume is changed, for the closed container, if the volume is changed, that means what happens? If the volume is changed, now the pressure also will be changed. Yes or no? This is the Boyle's law. Yes or no? In a closed container, if the volume is changed, now pressure will be changed in an inverse proportion, in the inverse relation. That means what happens? If the volume is increased, now pressure inside this will be decreased. If the volume is decreased, now what happens? Pressure will be increased. This is what happening for closed container. For the container, let us see. Now what happens? For the container, if the volume is changed, pressure is changed. And now the same will be exerted on the components that are present inside the container also. Please understand this. Suppose you are increasing the volume of this container. If you are increasing the volume of this container, what happens? Volume increase means pressure decreases. So pressure inside this container will be decreased. So the same will be exerted on the components that are present inside this also. That means if the pressure is decreased in the container, the lungs are present in the same container. So within the lungs also, the pressure will be decreased. So that means the changes in volume of lungs are in relation with changes in volume of this container called as thoracic cavity. So if the thoracic cavity volume is decreased, lung volume is also decreased. If the thoracic volume is increased, lung volume is also increased. So, what is the overall conclusion here? The overall conclusion is, since lungs cannot change their volume on their own, since lungs cannot change the pressure inside them on their own, they cannot change on their own. So that's why our body is changing the volume and the pressure of thoracic cavity. By changing the volume and pressure of thoracic cavity, the change in the volume and pressure of lungs can be done. This is what the relation. That's why during inspiration and expiration, you can feel that our chest is raising up and it is going down, raising up and going down. Why? Because changes in the volume of thoracic cavity will bring changes in the volume of lungs, means pressure inside the lungs will be changed. This is what the relation. You need to understand all these things with relation. What is the relation? Because these things are very, very important for assertion and reason type. Suppose you are simply by hearting the content, there is no use. If they are asking a logical question, then definitely will make a mistake. So don't make any mistakes here. Observe carefully. Once again, I am giving the conclusion because many students are making mistake here. That's why I am focusing on these points and repeating several times. Heart can undergo contraction and relaxation. That means it can change its own volume because it is having its own muscles called cardiac muscles. But lungs cannot alter or change their own volume and pressure because they don't have the muscles related to volume changes. That's why it is depending on the thoracic cavity. Thoracic cavity is an airtight container. Any change in the volume and pressure of thoracic cavity will show some changes in the lungs also. So that's why to create some changes in the pressures of lungs, so the thoracic cavity volumes are changed. And what is the overall relation here? Volume is inversely related to pressure. That means if the volume is increased, pressure inside it will be decreased. If the volume is decreased, pressure inside this will be increased. This is the relation, and I will link up this information with inspiration and expiration so that you can easily understand all these mechanisms. Clear? You don't need to by heart this. Understand this relation. So this is the significance of thoracic chamber. It is an airtight container. This is very, very important. Next one. So in this way, it can help in inspiration and expiration process where the thoracic cavity volumes are changed so that the volumes of lungs will be changed. So that the pressure inside the lung will be changed so that it will help in the exchange of gases. Anyhow, once again under the mechanism, once again I will tell you clearly. So please understand the relation here. Pressure and volume are inversely related with each other. Next one. So this particular differences in the partial pressure of gases is used for the exchange of gases at various locations. Clear? So this is what. And now let us quickly go through what are the various steps that they are talking here. What is the first step that they are talking here? Now these are the alveoli, that means lungs. Alveoli means what? They are the part of lungs only. These are the lungs, and here exchange of gases. This is the blood circulation, and these are the tissues. Yes or no? And here what exactly happening here? Now here oxygen rich atmospheric air is taken into the lungs. It is called as inspiration. And CO2 rich alveolar air is released outside the body into the atmosphere, and this is called as expiration. And this is what? That means exchange of gases, the movement of gas is taking place between atmosphere and the lungs. That is the first step they are talking here. Breathing is also called as pulmonary ventilation. Breathing is also called as pulmonary ventilation, by which atmospheric air is drawn in, it is taken into the lungs, and CO2 rich alveolar air is released out. This is the first step they are talking. And what is the second step they are talking? So oxygen from the alveoli will enter into the blood, and CO2 from the blood will enter into the alveoli. What is the in simple sentence? Exchange of gases like oxygen and CO2 are taking place across the alveolar membrane. This is what they are talking here. So diffusion of gases. So this exchange is by simple diffusion only. From higher partial pressure to lower partial pressure they are going across the membrane. That's why it is called simple diffusion. So diffusion of the gases like oxygen and CO2 across alveolar membrane is taking place. And once oxygen is entering into the blood, now it is going to the heart, and from there it is supplied to the tissues. So in this way oxygen is transported in the blood. Next one. From the tissue CO2 is entering into the blood, and now this CO2 is going to the heart, from there it is going to the lungs. In this way, the next step we are talking is the transport of gases like oxygen and CO2 by the blood. So that's why transport of gases by the blood. And the next point we are talking is oxygen from the blood is going into the tissues, and in this tissue CO2 is produced, and that CO2 is going into the blood. So that means exchange of gases like oxygen and CO2 is occurring at the tissues, at the level of tissues it is taking place. So diffusion of oxygen and CO2 between blood and tissues is the next important point. Next one, last one is utilization of oxygen by the cells. So now this oxygen is utilized by the cells, where? In that electron transport system. And then release of CO2, where? One standard example is in the Krebs cycle. Yes or no? In this way, cells are utilizing oxygen and they are releasing CO2, and this is called as cellular respiration, and these things are also part of this one. In this way, total 5 steps are there, but the last step, fifth step, is actually studied under a separate chapter called as respiration in higher plants, but remaining 4 steps we are studying in this particular chapter. Clear? So this is what another important information in a sequential manner, very clearly they mentioned in NCERT, so please try to remember this. Next one we are talking is regarding the mechanism of breathing. Mechanism of breathing is very simple thing, but many students are trying to by heart this, so that's why they are making mistakes. So I will tell you with a logical explanation so that you can easily understand this. So in this breathing process there are 2 steps. One is we are taking oxygen rich atmospheric air into the lungs, means into the alveoli, and that process is called as inspiration or inhalation. Generally it is taking approximately 2 seconds time. Next one, CO2 rich alveolar air is released from the lungs into the atmosphere, and this step is called as expiration or exhalation, and generally it is taking approximately 3 seconds. So for one complete cycle it is taking approximately 5 seconds. 5 seconds time. So in this way, in 1 minute total 60 seconds are there. 60 seconds divided by 5 times 5 seconds, you are getting 12 times. So on an average, if you are calculating in 1 minute 12 times we are doing this breathing, but general range is between 12 to 16. Accordingly these values are slightly variable. Clear? This is what about the calculation purpose. Next one, now we are going with the mechanism of breathing. For this mechanism of breathing, what is the role of this pressure differences? Please remember this. This mechanism is very important. Observe carefully. Always air moves from high pressure to low pressure. Remember this. High pressure to low pressure. Low pressure. This is the concept. In the entire breathing mechanism, you need to remember this one. Air always moves from high pressure to low pressure. If you are observing for oxygen, always observe where it is high, where it is low, where it is high, where it is low. Like that only, just observe, you will get the complete concept. If you are focusing on the CO2, for CO2 you just observe where it is high, where it is low. Always these gases are moving from high partial pressure. If you are talking for individual gas, in a mixture of gases, many are there. If you are talking for partial pressure of one particular gas out of many, it is called as partial pressure. If you are talking for individual gas, yes, high pressure to low pressure. Overall, if you see, overall pressure of the air, you just see from high to low. So always it is from high to low. Please understand this. Air always move from high pressure to low pressure. If you are talking for individual gas, then you need to say from high partial pressure to low partial pressure. There also it is from high to low only. So this is the basic logic you need to understand. So before you are understanding this mechanism, this is the point you need to understand. Air always moves from high pressure to low pressure. This is one important point. Second important point you need to understand is the relation between volume and the pressure changes. They are inversely related. If volume increases, pressure decreases. Volume decreases, pressure increases. If you know these 2 points, you can easily understand the mechanism of breathing. Now slowly one after the other we will go through here. So now let us say, first we will talk about inspiration. Inspiration. Now let us understand this. What exactly happens? Don't worry about the diagram. First I will explain the mechanism, then we will see the changes in the inspiration. What is inspiration? From the atmosphere, air should come into our lungs. Yes or no? From the atmosphere, air should come into our lungs. Now you just see, from where to where it is going? From the atmosphere, it should come into the lungs. From atmosphere to the lungs. That means where the pressure of the gas is high, in the atmosphere? In the atmosphere, the pressure is high. Inside the lungs, pressure is less. That's why it is coming from high to low. Yes or no? So during inspiration, what should happen? The pressure inside the lungs is less when compared with atmosphere. Yes or no? This is what we are doing. And next one, during expiration, what happens? From the lungs, air should go into atmosphere. So here there are two components. One is regarding the lungs. I am talking for one lung. Okay, this is the lung and this is atmosphere. This is atmosphere. Now during inspiration, what happens? During inspiration, from the atmosphere, it is coming into the lungs. This is inspiration. Yes or no? Next one, during expiration, what happens? From the lungs, it is going into atmosphere. This is what happening between atmosphere and lungs. Yes or no? No, very good. Now the thing is, what you are saying? During inspiration, atmospheric pressure is high and here it is low. That's why it is moving from high to low. But during expiration, you are saying that here it is high and here it is low. Understood or not? This is what happening. That's why you are releasing this air into the atmosphere. This is what the mechanism is. But the thing is, are both the values are getting changed? Are both the values are getting changed? Or the values of only one is getting changed? This is another important point. Please remember, I am a human being, so I am doing this breathing. That means inspiration, expiration. Now the point here is, can I change the pressure of atmosphere? Can I change the pressure of atmosphere? No. So now this is a constant. Now this will be a constant value. This is always constant. Then what actually you are changing? You are changing the pressure inside the lungs only. This is constant. I will give one rough example. Let us take some values here. I am taking some random values. Suppose the value for atmosphere is 10. Suppose this 10 will remain same only. For the lung, if it is becoming less than 10, less than means let us take 5 suppose. Now what happens? This is high, this is low. Now what happens? From the atmosphere it will come into the lung. Yes or no? Suppose inside the lung it is becoming 15. Now it is becoming higher than this. Now it will go like this. Understood or not? Some random values I am giving. These values are not related to the original information. This is just for example I am telling you. Now what is happening? This value will remain same only. That means we are not changing atmospheric pressure. What actually we are doing? We are actually changing the pressure inside the lungs. Pressure inside the lungs. Inside means intra. Lung means pulmonary. Intra pulmonary pressure. Intra pulmonary pressure. Inside the lungs, pressure will be changed. We are not changing the pressure of atmosphere. So simply by changing the pressure inside the lungs, inspiration, expiration can be done. Understood or not? So this is what the ultimate thing you need to do. Now the question is, then how you will change the pressure inside lungs? How you will change the pressure inside the lungs? Do lungs have any muscles for changing the pressure on their own? No. Lungs cannot change the pressure on their own. Yes or no? Now we will talk about, please remember all the things we are talking with respect to pressure. So now we will talk about inspiration. During inspiration, so what should be the pressure inside the lungs? Pressure inside lungs. Pressure inside the lungs. What should happen? Observe carefully. During inspiration, from the outside it should come. So what should be the pressure inside the lungs? It should be less than the atmospheric pressure. Lesser means negative. It is the negative pressure. Negative means it is lesser with respect to atmospheric pressure. But during expiration, it is a positive. Positive because it is higher than atmospheric pressure. So how you will create lesser pressure here? Negative pressure here. Negative sense means lesser than atmospheric. Some students what they are thinking is negative means it is negative value. It is not with respect to value. Here they are saying that it is lesser than atmospheric. That's why it is negative pressure. Higher than atmospheric. That's why it is called a positive pressure. In this way they are using these names. That's it. How you will create lesser pressure? We need lesser pressure. What is the relation between pressure and volume? Pressure and volume inversely related. You need less pressure. You need less pressure means what should happen to the volume? Volume should be high. Understand this. Each and everything is based on these two formula only. Please observe carefully. Pressure differences and the relation between pressure and volume. Inside the lungs, it should be less pressure. Less pressure means what we need? High volume. High volume of lung. Please remember this. High volume of lung. Lung volume should be more. How the lung volume will be more? If the thoracic volume is more, thoracic cavity volume is more. I am using up arrow for high and down arrow for low. Okay, thoracic cavity volume should be more. So how you will increase the thoracic cavity volume? This is the thoracic cavity, right? If the thoracic cavity volume is high, then the volume of lung is also high. If the volume of lung is high, pressure inside this will be low. If the pressure inside this is low, automatically air will come from outside to inside the lungs. That is called inspiration. So during inspiration, what we need to do? Pressure inside the lungs should be decreased. So to decrease the pressure inside the lungs, what we need to do? Thoracic means lung volume, pulmonary volume should be more. When pulmonary volume will be more? If the thoracic cavity volume is more. Now what you need to understand? How you will increase thoracic cavity volume? First thing is anterior-posterior side. So generally this is initiated with the contraction of diaphragm. Diaphragm under the relaxed state, it will be like a dome shaped. Relaxed state, it will be dome shaped. But here what we need to do? Please observe this carefully. So it is like this, right? So it is in the relaxed state. But now during here what should happen? Now here it is pressure like this, means volume is less. But what we need? We need more volume. How the volume will be increased? Now it should become flat. That means it should, I am changing the color here. It should contract like this. It is contracted like this. Now what is happening here? Volume is increased. Volume is increased. That means during this inspiration, what should happen? For the inspiration to occur, first phrenic muscles of diaphragm will undergo contraction so that it is trying to become flat. So antero-posteriorly the volume is increased. Nice. Next we are talking about dorsoventral. In the normal condition, what happens? So now these intercostal muscles are there. Now please understand, external intercostal, outside external intercostal, they are contracting like this. So when external intercostal muscles contract, what happens? Dorsoventrally the volume will increase because external intercostal muscles are contracting on outer side. Contraction will cause lifting of ribs and lifting of sternum. So now what happens here? Volume is increased. So in this way, thoracic cavity volume is increased. Please observe carefully. Once again I am repeating. For inspiration to occur, pressure inside the lungs should decrease. To decrease pressure inside the lungs, volume inside the lungs should increase. To increase volume inside the lungs, thoracic cavity volume should increase. How thoracic cavity volume is increased? By contraction of diaphragm. So that antero-posteriorly volume is increased. Yes or no? Antero-posteriorly volume is increased. Now next one is dorsoventrally. Contraction of external intercostal muscles will cause lifting of ribs and sternum. So dorsoventrally volume is increased. Now what happens to overall volume? Now overall volume is increased. For the thoracic cavity, overall volume is increased. Now the same is exerted on lungs also. Now what happens to the lungs also? For the lungs also the volume is increased. If the volume is increased, what happens? Pressure inside the lungs will be decreased. Pressure inside the lungs is decreased means what? Air from the outside, from the atmosphere will enter into the lungs. And this process is called as inspiration. That's it. Simple information. You don't need to by heart. Understand these relations. You will understand it very easily. So here you are talking about diaphragm, phrenic muscles and external intercostal muscles. In your syllabus, you are talking about only these. Yes or no? Contraction of these two muscles will cause increase in thoracic cavity volume. Increase in thoracic cavity volume will cause increase in pulmonary volume. Increase in pulmonary volume will cause decrease in pulmonary pressure. That means negative pressure is created. So air from atmosphere will be taken into the lungs. This process is called inspiration. This is the mechanism. Now you do exactly opposite for expiration. For the expression, what you need? Reverse. What is the reverse? Pressure inside the lungs should be more. Now reverse. If more pressure is required, less volume of lung is required. If the less volume of lung is required, less volume of thoracic cavity is required. So reverse. So thoracic cavity volume should decrease. How thoracic cavity volume will decrease? Reverse. Relaxation of diaphragm, relaxation of phrenic muscles will cause once again dome shape. That means antero-posteriorly volume is decreased. Next one, external intercostal muscles are contracted. So dorsoventrally volume is decreased. No, overall volume is decreased. Volume decreased means volume of thoracic cavity decreased means volume of lung will decrease. Volume of lung decreases means pressure inside the lung will increase. Pressure inside the lung increased means now expiration will occur. Simply opposite. You just remember anyone. You just remember anyone, but remember with clarity. You should not make any mistake. Remember any one of these process with complete clarity and logic and mechanism, so that exactly opposite is for the other type. So in this way, expiration and inspiration are taking place. It is a complete logical mechanism. Please don't try to by heart this, try to understand this. Clear? Pressure differences are important, and then relation between volume and pressure are important. So inspiration, thoracic volume is increased. Thoracic volume is increased means what? It will cause increase in pulmonary volume. So intrapulmonary pressure will be decreased. So it is negative pressure. So from the atmosphere, air will enter into this. And what is required? What is happening for this one? Diaphragm will contract. So instead of dome shape, now it will try to become flat. And then external intercostal muscles will contract so that ribs and sternum will be raised. So overall volume is increased. This is what the diagram is. That's it. Very, very simple mechanism. And expression exactly opposite. Now relaxation of the phrenic muscles of diaphragm will bring back dome shape. Once again dome shape. And the relaxation of external intercostal muscles. Now dorsoventrally the volume is decreased. Now thoracic volume decreased means volume inside the lungs is also decreased. Volume inside lungs decreased means pressure is increased. So positive pressure. Now air from lungs will go into the atmosphere. This is called expiration. Exactly opposite things. Now in NCERT they added one more sentence. The muscles of the abdomen and also internal intercostal muscles. Till now we studied only external, right? Internal intercostal muscles and the muscles of abdomen are also involved in some extra volume changes. And those details are not much important at this level. In higher classes you will study about those things. Clear? Next one is regarding respiratory rate. Just now I said no, for inspiration 2 seconds, expiration 3 seconds, this is the average value. So for one complete process it is taking 5 seconds. 5 seconds. For 1 minute total 60 seconds are there. 60 divided by 5 seconds. So 12 times breathing. So what is the respiratory rate? On an average it is 12 times per minute. But in NCERT they give 12 to 16 because these seconds values are sometimes variable in different individual. So average value is 12 to 16 times per minute. Clear? So this is what the value they are saying. And during this particular respiration or breathing process, how exactly these volumes are changed? How much air is taken in, how much released out, all these things we can measure or calculate with the help of an instrument called as spirometer. So there are some normal spirometers are also there, and digital spirometers are also available. So according to our requirement we can use this. Clear? And after completion of this mechanism, now we are talking about some respiratory volumes and pulmonary capacities. Respiratory volumes and pulmonary capacities. So roughly also we can represent like this. By adding various volumes, we can get different capacities of lung. Clear? So let us quickly go through one by one. First one is the tidal volume. Under normal inspiration, R. Please remember they have given a word called R. Many students they are thinking that it is und. It is not und, it is R. Please remember. You just calculate during inspiration. R. Calculate during expiration. Any one only you just take it. Any one you just take it. So in the normal inspiration or in the normal expiration, what is the volume of air that is taken in, or volume of air that is released out? That normal value of that volume of air is called as tidal volume. So for an healthy individual, it is approximately 500 ml. Since we are taking the breaths for 1 minute, if you are calculating 12 to 16, 12 to 16. So 12 into 500, to 16 into 500. So it is approximately 6000 to 8000 ml. So if you are converting into liters, 6 liters to 8 liters in 1 minute. Please remember, only tidal volume we are calculating for 1 minute. Remaining volumes we are not calculating for 1 minute, because every time we are not doing this. Very rarely we will do these things. So that's why per minute calculation we are doing only for tidal volume, not for other volumes. Please remember. Inspiratory reserve volume, anyhow tidal volume we are taking. If you are doing forced inspiration, some extra volume you can take it. That extra volume that we can take during this forced inspiration is called as inspiratory reserve volume, and its volume is approximately 2500 to 3000 ml in one time itself. This is calculation for one time itself. Clear? Next one, expiratory reserve volume. Anyhow we are releasing the tidal volume. So upon forced expiration, some extra volume of air we can release. That extra volume we are releasing during forced expiration is called as expiratory reserve volume, and its value is approximately 1000 to 1100 ml. Next one, residual volume. Even after forced expiration also, some volume of air is present inside the lungs. That is called as a residual volume, and it is approximately 1100 to 1200 ml. Clear? And in this way, these are the various volumes. By adding these various volumes, we can get some pulmonary capacities. First, we are talking about inspiratory capacity. What is the inspiratory capacity? Anyhow, we can take the tidal volume. Along with this, in forced inspiration, we can take inspiratory reserve volume. So when you are adding these two, you will get inspiratory capacity. Next one is regarding expiratory capacity. Anyhow, we can release the tidal volume. During forced expiration, we can release expiratory reserve volume. By adding these two, we can get expiratory capacity. Next one is functional residual capacity. Under normal condition, what is the air that is present inside our lungs? Anyhow, residual is present. Along with this, expiratory reserve volume is also present inside the lungs under normal condition. So that's why these two combinedly it will form functional residual capacity. Next one is regarding vital capacity. You can measure this in two ways. Okay, forced expiration followed by forced inspiration, like this you can do. Reverse, or forced inspiration followed by forced expiration, like this also you can calculate. Anyone you can calculate. So in this what will come? Inspiratory reserve volume, tidal volume, expiratory reserve volume. All the three combinedly they are making this vital capacity. Okay. Next one is regarding total lung capacity. Along with this vital capacity, if you are adding that residual volume also, so what is the total volume of air that is present inside the lungs after forced inspiration? So that means all the four will come. Residual volume will come, expiratory reserve volume will come, tidal volume will come, and inspiratory reserve volume will come. So all these four combinedly they will give this total lung capacity. And now these things you can write in a different way. So once you understand this, you can write them in any way. So please understand this. In NCERT they gave that total lung capacity is all these four, addition of all these four. Or you can write this as, here they gave that vital capacity plus residual volume. Only these two they gave in NCERT. But now since you know these relations, now you can write the same thing in a different ways, So that's why please remember, practice all these things. You just think on it, think on your own, and how in different ways you can write all these things. Practice this. The same things, you can interchange them. So that's why for the examination purpose, it is not just only the two they have given in NCERT. Remaining things also you should be able to write. Suppose these are the four. Residual volume plus expiratory reserve volume. It will form functional residual capacity. And these two as it is we are writing. Suppose tidal volume plus expiratory reserve volume. It is called expiratory capacity. And these two you just write as it is. And tidal volume plus inspiratory reserve volume is inspiratory capacity. These two you just write as it is. Yes or no? And then residual volume keep separately. These three you just add. You will get vital capacity. Yes or no? And these two combinedly functional residual capacity. These two combinedly inspiratory capacity. So in any way you can write it. So once you understand this, you should be write these in different ways. And if you still have any doubt, you can pause the video and you just note them down in the textbook itself. This is for total lung capacity. And this is how you can write the vital capacity in different ways. I don't want to explain all these things. I hope you understood how to write this. Next one is functional residual capacity. You can write like this. And expiratory capacity, you can write in these different ways. Clear? So next one, inspiratory capacity, like this. whatever it is. So once you understand one concept, you should be able to write them in different ways also, and with that, that particular concept is complete. Now we are talking about transport of the gases. Transport of the gases. Please remember, for the transport of the gases, there are three important things. There are three important things that are present, and we need to understand all these things. First important one is regarding the partial pressure of gases. Partial pressure of gases. First one, for the transport of gases, for the movement of gas from one location to other location, first important one that is playing significant role is the partial pressure of that particular one. If the partial pressure difference is very high, quickly it can move from one location to other location. So that's why it is very, very important. Next one is regarding the solubility. Solubility of gases. Solubility of the gases. Since we are talking about solubility of these gases, blood is helping in transport of this. Blood is nothing but mainly it is the water, right? So if these gases are more soluble in the blood, more soluble in the water, now they can be quickly transported. So please understand this. If the solubility is more, they can be easily transported. Next one. Next one is regarding thickness. Thickness of diffusion membrane. Thickness of diffusion membrane, very, very important one, because at the lungs and at the tissues, transport of gases means a diffusion of gas is taking place. So at that diffusion membrane, what is the thickness? If it is very thin, if it is very, very thin, now the diffusion will be very quick, fast. And if the membrane thickness is very high, diffusion will be very less. So that's why these are also some important points that are helping in transport of these gases. So you need to observe this concept very carefully. Now we will go with one after the other very clearly. Transport of the gases. What are the gases that we are talking here? Only two gases we are focusing. One is oxygen and the other one is carbon dioxide. With respect to transport of oxygen, if we see, 97% of oxygen is carried by RBCs. Within the RBCs, hemoglobin is present. To the hemoglobin, oxygen will attach and it will form oxyhemoglobin. So that's why 97% of oxygen is transported as oxyhemoglobin. But remaining 3% is directly dissolved within that blood. So in the dissolved form, it will be transported. This is what they are saying. And with respect to carbon dioxide, if we talk, 7% is in the dissolved form it is transported. In the remaining one, 70%, that means it is the major fraction, right? 70% is transported as a bicarbonate. Mechanisms also we will study. And the remaining 20 to 25%, it is going through the RBCs. RBCs means again in the form of this attachment with hemoglobin. But CO2 attached with hemoglobin, it will form carbaminohemoglobin. Many students are making mistake here. They are writing carboxy. It is not carboxy. When CO2 is attaching with hemoglobin, it will form carbaminohemoglobin. It is not carboxyhemoglobin. Okay. Carbon monoxide will form carboxyhemoglobin, but that is not in your syllabus. So please remember this. So these type of some extra informations, but still linked with this. So I have covered under extra lecture in the regular chapters, in the regular lectures. So those who are interested, you can go through the regular courses that are available on the app. Next one, transport of oxygen. Now first we will talk about transport of oxygen. First one is regarding dissolution of the gases. We are saying that 3% of oxygen is in the dissolved form, right? Now understand here, this is the basic general information. For a particular gas, if the partial pressure is high, more molecules will be dissolved within this particular liquid. But if the partial pressure is less, the number of molecules that are dissolved within that liquid will be less. So in this way, you can understand that if the partial pressure is more, the dissolution rate will be more. Clear? So in this way, this is one general information that you need to understand. And with respect to oxygen, if you talk, 97% it is transported in the form of oxyhemoglobin and only 3% is transported in the dissolved form. In this way, oxygen will be dissolved in this particular liquid and along with this it is going. So this is called as dissolved form. And now we will talk about this oxyhemoglobin. Before going with oxyhemoglobin, first we will talk about hemoglobin. So hemoglobin molecule is having the heme group. heme group, and in this heme group at the center it is having iron in the +2 oxidation state. Please understand this. So in the case of hemoglobin, iron is having Fe+2 form. Very, very important point for examination. Clear? And then how many chains are present in this hemoglobin? Please remember, at our level we are talking about hemoglobin adult 1 type. Hemoglobin adult 1 type. Regarding these particular things, just some small information I have given you when we studied about biomolecules. So originally we are talking about hemoglobin adult 1 type, hemoglobin adult 2 type, hemoglobin fetal type. At least for examination you need to remember these two. Clear? And in these hemoglobin types, whatever is the hemoglobin type, four chains are present. Out of these 4 chains, 2 chains are alpha forms. 2 are alpha forms. Here alpha form, beta form, right? So what is that beta form concept applicable? Where exactly it is applicable? Only for adult 1 type. In all the 3 alpha chains are present. How many? 2 alpha chains are present. But beta chains are present only for hemoglobin adult 1 type. They are not present in other types. In hemoglobin adult 2 type, delta chains are present, but not mentioned in NCERT. Hemoglobin fetal type, it is having gamma chains, not mentioned in NCERT, but these two are important for examination. Please remember this. Now, generally we are talking about alpha to beta 2 forms because these two are adults. These are in the fetal. Please remember, when the fetus present inside the mother's womb or the uterus, then what exactly is present? Fetal type of hemoglobin is present, not this. This type is present. And this type of hemoglobin has more affinity to bind with oxygen. When we study about oxygen dissociation curve, I will tell you relation with interlinking also. This is very important concept. Even though not mentioned in NCERT, it is very important to understand. Clear? I will tell you about this. This is the fetal type, present only in the fetus. But now here, after the birth, what happens? Now slowly it will be replaced with adult type. In the adult type also, HbA1 type is about 97%. That's why always we are talking about alpha to beta 2. But the HbA2 is just 3%. So even though you are not studying this, there is no problem. So that's why we are focusing on this particular one. Regarding this, I will tell you separately under oxygen dissociation curve. Now here let us come back to this one. Alpha to beta 2. That means it has 2 alpha chains and 2 beta chains. You need to interlink this information with thalassemia and sickle cell anemia concept that we are studying in principles of inheritance and variation. Sickle cell anemia is due to a mutation in beta chain gene, and it is a qualitative problem. But thalassemia is a quantitative problem. If alpha chain is not produced, now it is called alpha thalassemia. If beta chain is not produced, it is called beta thalassemia. But in sickle cell anemia, beta chain is produced, but it is not normal. It is a mutated beta chain. Quality is compromised. That's why it is a different type. So these two informations also you need to interlink. And in principles of inheritance and variation, very clearly they have mentioned this. Alpha chain genes are present on chromosome number 16. Beta chain genes are present on chromosome number 11. For the alpha chain, there are 2 genes. So 2 into 2, 4 alleles are present. For the beta chain, 1 gene is present. That means 2 alleles are there. So this information was very clearly mentioned in principles of inheritance and variation. You just link up that information here also. Next important information. Hemoglobin is having 4 chains. All these 4 chains are interlinked with each other through the heme group. Since all these are interlinked, now this protein structure, if you see, what is the level of organization that you are studying? It is the quaternary structure. In the biomolecules, for the quaternary structure of protein, we are studying about hemoglobin example. Link up all these things. This is what the information that you need to understand. Clear? With this, the basic information is completed. This is what the quaternary structure. In this way, it is having the protoporphyrin ring inside this. Protoporphyrin ring, or simply it is the heme group that is present in this. And in this heme group, Fe is present in +2 form. Please at least remember this information. Clear? Now we will talk about oxyhemoglobin. In this case of oxyhemoglobin, please remember, so in one hemoglobin molecule, total 4 heme groups are present. One hemoglobin has 4 heme groups. For each heme group, now here to each iron, now one O2 molecule is attached. So 4 heme groups, so 4 irons, so O2, 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 O2. So total how many oxygen molecules are attached in one hemoglobin? So one hemoglobin will attach to 4 oxygen molecules, don't make any mistake here. So here one hemoglobin will bind with 4 oxygen molecules. 4 oxygen molecules means how many atoms? So 8 atoms, 8 atoms of oxygen. This question is very important. Many students are making mistake. 4 molecules of oxygen. So read the question very carefully. Are they asking about molecules of oxygen or atoms of oxygen? If they are asking about molecules of oxygen, answer will be 4. If they are asking about atoms of oxygen, answer will be 8. 8. So please observe these. You know all the concepts. But if you don't understand the difference between these words, you will make a mistake. So please try to understand, read, understand these questions. There is no hurry in this. Okay. This is what. Now in this way it is attached. Next one, here oxygen is attached to this iron, right? So oxygen is attached to this iron. So now this particular process is it oxidation process or oxygenation process? This is also another very important word. Many students are making mistake here. So generally what is oxidation? Oxidation is the attachment of oxygen or removal of hydrogen or removal of electrons. This is how you are giving the definition of oxidation. Here what you are saying, oxygen is attached to this iron. So will you call this as oxidation? This concept is very important. Please remember, in this case what we are saying is, when oxygen is not attached, iron is present in Fe+2 state. When oxygen is not attached, in the hemoglobin, iron is present in Fe+2 state. And when oxygen is attached, then also it is in Fe+2 state. It is not oxidized to Fe+3. Please understand this. If Fe+3 is present, that is called methemoglobin or methemoglobin. That you are not studying here. Please observe carefully. In the oxyhemoglobin, when oxygen is attached to hemoglobin at the iron, oxidation state of iron is not changed. It still remains as Fe+2. So that's why it is not an oxidation process. It is not oxidation process. It is simply attachment of oxygen. That's why this is called as oxygenation process. Please understand the difference between these words. Attachment of oxygen to hemoglobin is oxygenation process. It is not oxidation process. Why? In the assertion reason they may ask why? Because oxidation state of iron is not changed. Very important for examination. Try to understand these basic informations. Clear. Next one we are going with the next one, that is called as oxygen dissociation curve. What exactly we are understanding from this oxygen dissociation curve? Please remember one thing, this graph we are getting in an experiment. In the laboratory, we are doing this particular experiment, and based on the experimental value, we are making this particular graph. This is the first thing you need to do. Many students are not understanding what is the significance of this graph. If you don't understand the significance of this graph, if you can't use this graph for calculation purpose, that means you did not study this graph well. So please observe this carefully. Since many students are making mistake on this, I will try to spend little bit more time on this because remaining informations are very simple. Let us try to understand this very clearly. How exactly you need to do mathematical calculation. If they are giving some questions, that also I will tell you. Don't worry. So observe very carefully. So in the experiment, what you are doing, you are using some various partial pressures of oxygen. So this is 10 mm of mercury, 20 mm mercury, 30 mm mercury, 40 mm mercury, 50, 60, 70, 80, 90, 100 mm mercury. So in this way you are using different partial pressures. And at these different partial pressures, what exactly you are calculating? Percentage saturation of hemoglobin with oxygen. That means oxyhemoglobin you are measuring here. So how much of hemoglobin is interacting with oxygen? That means how much oxyhemoglobin is formed. percentage saturation. So 10% formed, 20% formed, 30, 40, 50, 60, 70, 80, 90, 100% formed. So in this way, on the y-axis we are taking percent saturation of hemoglobin with oxygen. On the x-axis we are taking different partial pressures of oxygen. This is an experimental setup. So when they have taken 10, they got this value, and when it is 20, they got like this 30, it is like this 40, it is like this 50, it is like this 60, 70, 80. So they got different values and they have attached this. So once they have attached this, what is the shape of this particular graph? Very important question. What is the shape of this? It is a sigmoidal. It is a S-shaped, sigmoidal shape. This is the first important point. And in this way, they made one standard graph. And how exactly you can use this graph with respect to human condition? With respect to human condition. Now I will try to link up this information, try to understand this. Clear? Now let us see. In this way, they are making this standard graph. Nice. And this standard graph we need to link up with. This point I will tell you separately, don't worry. So this is the NCERT diagram that they have given. And here we are talking about exchange of gases. So transport of gases. Please remember, always these gases are moving from high partial pressure to low partial pressure, yes or no? First thing, partial pressure we are talking. Now observe carefully. In the NCERT they gave table also. I think I have not given the table. So in the atmosphere, what is the partial pressure of oxygen? 159 mm mercury. So CO2 is 0.3 mm mercury. Likewise, at different levels they have given. No? I will tell you this. In the atmosphere, what they have given? 159 mm mercury. In the alveoli, I am talking only for oxygen. Only for oxygen. We will focus on only oxygen. Clear? So now better will be, I will explain. Don't worry. So first let us see about atmosphere. First we will see about partial pressure of oxygen. In the atmosphere they have given 159, all these values in mm mercury. Okay, 159. And then from the atmosphere it is coming into the lungs. Lungs means alveoli. In the alveoli, what is the value they have given? It is 104 they have given. Yes sir, 104 mm mercury they have given. And then what we are talking? So now it is going into deoxygenated blood. Deoxygenated blood. So in the deoxygenated blood, how much it is? It is 40 mm mercury. Once oxygen from alveoli is entering into deoxygenated blood. Now since oxygen is entering into this, now it will become oxygenated blood. Now it will become oxygenated blood. In the oxygenated blood, it is 95 mm mercury. Here they have given 95 mm mercury. Please observe all these values. 95 mm mercury. Now this oxygenated blood is going to the tissues, but inside the tissues it is 40 mm mercury. Please observe carefully here. From where it is going here, from where to where it is going, always it is moving from high partial pressure to low partial pressure. From the atmosphere it is coming to the alveoli, higher to lower. From the alveoli it is going into deoxygenated blood. What is the partial pressure in deoxygenated blood? It is 40 mm mercury. It is going. So since oxygen is continuously coming and coming, now it will become oxygenated blood. It is 95 mm mercury. Now this oxygenated blood is going to the tissues. Now from the 95, it is going into the 40 mm mercury. So please observe in all these cases. In all these cases, oxygen is moving from higher partial pressure to lower partial pressure. Observe this. Observe this. This is what the information you are studying for oxygen. Now we will see for the CO2. For the CO2 we will see here. For CO2, here also it is in mm mercury. But it is reverse, right? So CO2 is more produced in tissues. CO2 is more produced in tissues. And here what is the partial pressure? It is 45 mm mercury. Entire information was given here. For the tissues it is 45 mm mercury. And then from the tissues it is going into oxygenated blood. Oxygenated blood. In the oxygenated blood, what is the partial pressure of CO2? It is 40 mm mercury. That means tissues into oxygenated blood it is going. Since CO2 is entering into oxygenated blood, after some time what happens? It will become deoxygenated blood. Deoxygenated blood. Deoxygenated blood. Since CO2 is entering more and more into oxygenated blood, it will become deoxygenated blood, and here it will get 45 mm mercury. And now this deoxygenated blood is coming to the alveoli. And now here it is going into the alveoli. Inside the alveoli it is only 40 mm mercury. 40 mm mercury. Yes or no? And then from the deoxygenated blood, now the CO2 is entering into the alveoli. And from the alveoli, it will go into the atmosphere. There it is 0.3 mm mercury. Understood or not? Now observe very carefully. From where to where it is going? From the tissues to the oxygenated blood it is going. Higher to lower. And since CO2 is entering into oxygenated blood, it will become deoxygenated blood having 45 mm mercury. Same deoxygenated blood is going to the lungs. At the lungs, now here from higher to lower it is going. That means it is diffusing from the deoxygenated blood into the alveoli. And during expiration, now it is going from the alveoli into the atmosphere. Yes or no? You just observe here. Now it is simply reverse direction. Now in this way, oxygen from the atmosphere is coming into the tissues and there it will be utilized and CO2 is produced. And the CO2 is partial pressure is more in the tissues, so that now it is ultimately released into the atmosphere. So please remember all these values you should remember like this, sequence wise, from where to where they are going. So in this way you can understand that how oxygen is transported, how CO2 is transported. This is what the basic important information is. Clear? These are the values, everything they gave. Not only this, in the NCERT they gave a separate table also. I gave all the values. Table also they gave. Remember that. You need to remember any information given in NCERT. You need to remember it. Clear? This is what they are saying. And here our aim is for the oxygen transport. And please remember this hemoglobin. This is with hemoglobin. Where hemoglobin is present? Is hemoglobin present inside this tissue? Not in these tissues. It is present within the blood. So not inside the alveoli. So hemoglobin concept we are not studying for alveoli and these tissues. We are not studying for alveoli and these tissues. Where exactly we are focusing? We are focusing within this blood. We are focusing within the blood. Now observe carefully. During inspiration, oxygen rich atmospheric air is coming into alveoli. And here exchange of gases is taking place here. Yes or no? And here there is a membrane here. So what is that membrane here? So here this membrane thickness is very less. Membrane thickness is very less. In NCERT they gave separate diagram also. So it is the simple squamous epithelium of alveoli and the simple squamous epithelium of blood capillary, that is also called endothelium. And simple squamous epithelium is very thin, and between this is a non-cellular basement membrane. And all the three combinedly, the thickness is around 1 millimeter. That means very thin. So since the thickness of membrane, diffusion membrane is very less, diffusion of gas is very quick. This is one important point. Yes or no? And the other point is partial pressures, higher to lower always going. Yes or no? Next one, solubility. So compared to oxygen, solubility is 20 to 25 times higher for CO2. So under the CO2 I will tell you separately. And here once this oxygen is coming into the blood, now inside the blood you need to follow this. And here what you are talking, on this side you are talking about oxygenated blood. For the oxygenated blood, what is the partial pressure? 95 mm mercury. Now this information you just use here. When the partial pressure of oxygen is 95 mm mercury, when the partial pressure of oxygen is 95 mm mercury, then what is the percentage saturation of hemoglobin with oxygen? This is the question. So 95 mm mercury where we are observing? In the oxygenated blood. This is 80, this is 90, this is 100, 90, 100, between 95, make one line, 95, and it is almost coming to 100%. As per NCERT diagram, if you are observing, it is almost coming to 100%, but originally it is around 98 to 99% saturated. Understood or not? What is the meaning of this? This particular graph you need to use here. That means in the oxygenated blood, what is the percent saturation of hemoglobin with oxygen? It is almost 100%. That means 98 to 99% hemoglobin is saturated, that is attached with oxygen. This is what the use of this graph here. Very good. So in this way, that hemoglobin is coming with more and more oxygen. It is reaching the tissues. At the tissues, what happens? It is giving its oxygen to the tissues. Different factors are there. Slowly we will study factors also. So different factors are responsible. So oxygen is separated from hemoglobin. So oxygen is separated means it is a dissociation of oxygen from hemoglobin. Once it is dissociated from hemoglobin, where it is going? It is entering into the tissues. There are some factors here. High partial pressure of carbon dioxide, high H plus ion concentration, high temperature, and then high values of 2,3 bisphosphoglycerate or 2,3 diphosphoglycerate. These are the factors that are seen at the tissues. Because of this, oxygen is easily dissociated from hemoglobin so that it can enter into the tissues. This is the information that you need to understand. Okay. Sorry. Sorry. Some amount of oxygen is released, but remaining oxygen is still attached with hemoglobin. How much it is? Come to this one. What is this blue part? Blue part is deoxygenated blood. Please remember, many students they are thinking that deoxygenated, D means removal, oxygen is removed. Many students they are making mistake. What is the mistake they are doing? They are thinking that deoxygenated blood does not contain oxygen. That is wrong. Deoxygenated blood, partial pressure of oxygen, some amount of oxygen is there. So please remember here, though it is deoxygenated blood, still oxygen is present. And here, though it is oxygenated blood, CO2 is also present. So oxygenated blood has both oxygen and CO2. Deoxygenated blood is also having both oxygen and CO2. Don't make mistake. Many students are making mistake here. So please observe carefully. In both the cases, both of them are there, but partial pressures are different. Oxygenated blood is having higher partial pressure of oxygen when compared with deoxygenated blood. For CO2, if you see, deoxygenated blood has more partial pressure of CO2 when compared with oxygenated blood. So, but in both the cases, both the gases are present. Partial pressures are different. Don't make mistake. Okay. So, in the oxygenated blood, since 95 mm mercury partial pressure is present, almost 99% of hemoglobin is attached with oxygen. But when you are talking about deoxygenated blood, in the deoxygenated blood, what is the partial pressure of oxygen? It is just 50 mm of mercury, sorry, 40 mm mercury. So for the 40 mm mercury, just make the line here. Where it is coming here? This is 60, 70, 80. So that means it is almost coming to 75% saturated. 75% saturated. Now I will give you link up. I will give you calculation. All these things were given in NCERT. Calculation was not given, and many teachers are not teaching this calculation part. So that's why students are trying to by heart the information, and when application question is given, they are making mistake. Now I will try to solve all these things in single stretch. So please try to remember this information. Observe how exactly I am doing this calculation. Observe carefully. Oxygenated blood. Oxygenated blood. In the oxygenated blood, what is the partial pressure of CO2? So partial pressure of CO2 is 95 mm mercury for oxygen. And there, what is the percent saturation of hemoglobin with oxygen? What is the percent saturation we are saying? It is almost 100%. It is almost 100% it is saturated. Clear? Next one. So in the case of deoxygenated blood, this calculation is very, very important. So observe carefully. In the case of deoxygenated blood, what is the partial pressure of oxygen? It is just 40 mm mercury. And when it is 40 mm mercury, just now we studied. So what is the value you are getting? 75% saturated. It is 75% saturated. 75% saturated. What is the meaning of this? What is the meaning of this? Oxygenated blood is having 100% oxygen, but deoxygenated blood is having only 75% oxygen. That means to the tissues, what is the difference between this? The difference between this is nothing but that is the oxygen given to the tissues. That is the oxygen, as per NCERT word, if you are giving oxygen delivered to the tissues. Understand the concept. This is the oxygen that is delivered to the tissues. So how much it is delivered? 25% oxygen is delivered to the tissues. I will give you the very, very important questions with answers here. So please observe carefully. 25% oxygen is delivered to the tissues. Percentage wise, it is okay. But in NCERT, they said that, what did they said? 5 ml oxygen is delivered to the tissues. That's what they gave. Now I will link up that information. Observe carefully here. I am linking these two things. Observe very carefully. 100 ml oxygenated blood. 100 ml oxygenated blood carries how much of oxygen? It is the 20 ml oxygen. Very clearly they mention NCERT. Yes or no? So actually in the not in this chapter, in the next chapter that is called body fluids and circulation, they said that 100 ml of blood will carry 20 ml of oxygen. Very clearly they mentioned in NCERT. 100 ml of blood carries 20 ml oxygen. And here 100 ml blood delivers how much of volume here? 5 ml blood to the tissues. In NCERT they mentioned this, but now here you need to understand this. 100 ml of deoxygenated blood carries, if this question is asked, what you need to write? 20 minus 5, it is approximately 15 ml. It is approximately 15 ml. How you are getting this? Now observe carefully. Anyhow you know that, because in this hemoglobin is there, so 1 gram of hemoglobin will carry so and so amount of oxygen. All these relations were given in the next chapter, that is body fluids. There I will give the relation. Observe carefully. 100 ml oxygenated blood is having 100% saturated oxygen. That means it is carrying 20 ml of oxygen. Nice. And then here, so here 20 ml is 100%. 20 ml is 100%. But out of this, what is the percentage that is released to the tissues? Only 25% is released. What is the 25% of 20 ml? It is the 5 ml. So 5 ml is delivered to the tissues. That means what is the amount of oxygen that is carried by deoxygenated blood? Still it is carrying 15 ml with it. Still it is carrying 15 ml with it under normal conditions. What are normal conditions? These are the normal conditions. Under normal condition, what you are talking? Deoxygenated blood is having 20 mm mercury. So under this condition, what you are saying? So only 5 ml. That means only 25% oxygen is delivered to the tissues. These are the calculation part. Remember this. Now I will give one application question. What is that application question? Suppose this is under normal condition. I am in the normal resting condition. So this is what the value is. Suppose I am doing some exercise. Under exercise conditions, what happens? Tissues will use up more and more oxygen. So when more and more oxygen is used up, so generally we are observing 20. Please understand this. Under the normal condition, 40 mm oxygen is present in deoxygenated blood. But under strenuous, excessive exercise conditions, generally we are observing 20 mm mercury in deoxygenated blood. If deoxygenated blood is having 20 mm mercury, then what is the amount of, or what is the volume of oxygen that is delivered to tissues? What is the volume of oxygen that is present within deoxygenated blood? I hope now you understood the concept. Now you need to do the calculation. Better you just pause the video, do the calculation, and then you just check it. Now I am continuing with the answer. Observe carefully this one. This concept is very important. There are some MCQ questions are also based on this. But students and teachers, many teachers, they don't know how to do this calculation. So please observe carefully. Understand the concept. Don't try to by heart this. So when it is 95 mm mercury, it is 100% saturated and 100 ml blood is carrying. 100 ml blood has 20 ml oxygen. Clear? But now what is our question? Under excessive exercise condition, I am giving that deoxygenated blood is not having 40, it is having 20. If they are giving 20, so we need to talk about 20 mm mercury. Under 20 mm mercury condition, what is the percentage saturation here? 20, you just draw the line here. Where it is cutting here? 30%. So 30% is saturated. It is 30% saturated. 30% saturated means how much it is delivered? Delivered is how much? 70% is delivered. Please observe this carefully. In the previous calculation, at the 40 mm is 75% saturated. So 100 minus 75, it is 25% that is delivered. But in this case, it is just 30% saturated. That means 70% oxygen is delivered. 100% is 20 ml means 70% is how much? If 100% is 20 ml, 70% is how much? It will be approximately 14 ml, right? It will be 14 ml. So 14 ml oxygen is delivered to tissues. Is delivered to tissues. Observe carefully. Then in the deoxygenated blood, how much of oxygen will remain here? 20 minus 14, that means it is approximately 6 ml. These are all approximate values. Sometimes in the options they may change these numbers. So whatever is the nearest number is there, that number you need to take it. So here it is, 6 ml oxygen is present in deoxygenated blood. Understood or not? So under normal conditions, 100 ml blood is delivering 5 ml of oxygen, but under very strenuous exercise conditions, more oxygen is required for the tissues. So that's why more and more oxygen is delivered. This is how you need to calculate these particular things, and you need to derive these answers based on this standard graph. This is what the significance of standard graph. If you don't understand this type of significance, that means you did not study this well. So please understand these logical interrelations also. In this way also some application questions can be asked, and these are also important for examination. So please understand the significance of this sigmoidal curve. It is not just randomly given. You need to interlink it with the information given in NCERT. What is its significance? And in the NCERT, at the end of the paragraph, they gave that 5 ml of oxygen is delivered to the tissues by 100 ml blood. And how they gave this value? This is what the relation. So these relations also you need to understand. Please don't make any mistakes, because if single mistake can cause 1 year delay, you may need to go for repetition once again. So that's why please, whenever we are studying, so study with clarity, so that you will never make any mistakes. So this is what, and this is how exactly we are doing calculations, and this is what the significance of this particular sigmoidal curve. This is the basic information. Now come to this particular one. And here what they are saying is, so this is the standard graph actually. This is actually the standard graph. But at the tissues, there are different conditions. At the alveoli, there are different other conditions. So in these conditions, how exactly this graph is changed? First I will tell you, suppose this middle black one is the standard graph, and when it will shift to left side, this is left side, and when it will shift to right side. So these significant factors also we need to understand. And before directly telling about the logical answer, first I will tell you how exactly you need to see the graph. Many students are facing difficulty in understanding the graph itself. So first what we will do is, suppose this is the standard graph. This is the partial pressure of oxygen, and this is the percentage saturation of hemoglobin with, this symbol is the with, okay, with oxygen. Percentage saturation of hemoglobin with oxygen. This is what we are taking. Now we will draw the standard graph. Suppose this is the standard graph. Suppose standard graph is like this. So this is under the normal experimental conditions, we have got this particular sigmoidal shape of the graph. But here what we are saying is, in some cases, it may shift to the left side. It may shift to the left side. This is called as a left side shift. It is called as left side shift. And in some cases, it may shift to the right side. This is called as right side shift. Right side shift. Center one is the standard graph. Standard graph. In comparison with standard graph, and here we are observing some left side and right side, two types, two shifts we are observing. We are observing two shifts, and we have two conditions. There are some conditions that we are observing at the tissues. There are some conditions we are observing at tissues, and there are some conditions that we are observing at the lungs, at alveoli. Lungs means alveoli. At the alveoli we are observing. What are these conditions? And why they are causing left side shift? Why they are causing right side shift? Directly conditions I can write first, but I will give the conditions later. First we need to understand what is the significance of this particular left and right side shift. What do they indicate? You need to compulsorily know about this. If you don't know this, again you will make some mistakes. So that's why what I will do is, this is the standard graph. This is the standard graph. First we will talk for standard graph. Here you are writing about the partial pressure. Yes or no? So here we will write about some 50% saturation. Suppose here for the standard graph, here you are getting suppose 100% saturated. 100% saturation. So this is a graph paper. So on this graph paper, for the 50% saturation, let us take this 50% saturation. So we were talking about 50% saturation. For the standard graph, let us see. For the standard graph, for the 50% saturation we are taking, and from there we are drawing a line onto this standard graph, and wherever it is touching, from there we are drawing a line onto the x-axis, so that it is touching to the x-axis. Clear? And for the standard way, standard 50% is having a particular value. 50% saturation you are drawing, so approximately it is coming around 27 to 28 mm mercury. Yes or no? 50% saturation, draw the line, and here you are getting, this is 20, this is 30, right? Before 30 you are getting, so around 27, 28 you are getting that value. I don't want to go into those details, so leave it. That is the standard P50 value. What is the standard P50 value? For 50% saturation, what is the value that you are getting? Suppose it is 27 mm mercury. This is the standard value you are getting. So this is called as P50. P50. What is the meaning of P50? Partial pressure of oxygen. Partial pressure of oxygen that is required for 50% saturation. For 50% saturation, how much partial pressure is required? That value is called as P50 value. This is the P50 value. But this is for standard graph. If it is having left side shift, what is the meaning of this? If it is having right side shift, what is the meaning of this? Okay. So if it is left side shift, same 50%, it is cutting here, draw the line here, draw the line here. Let us say this as X. Suppose it is right side shift, draw the line here. This is also 50% saturation, and draw the line onto this, and let us say this as Y, X and Y. Now observe carefully. What is the conclusion? How exactly you need to see this particular graph? I will tell you that. Observe carefully. Normally for the standard graph, for 50% saturation you need so and so, so and so. We are not worried about values. So and so partial pressure of oxygen is required. But if the graph is having left side shift, if the graph is having left side shift, for the same 50% saturation, what is this value? Higher than 27 or lesser than 27? This is less than 27, right? That means for the same 50% saturation, less partial pressure. That means even the less partial pressure of oxygen also is giving 50% saturation. What is the meaning of this? At the lesser partial pressure itself, 50% saturation is obtained. What is the meaning of this? In this case, hemoglobin has more affinity, more binding affinity with oxygen. That's why at lesser partial pressure itself, it is getting more 50% saturated. Understood or not? Once again, I repeat the sentence. Try to understand the concept. At the lesser partial pressure of oxygen itself, 50% hemoglobin is saturated. Understood or not? That means in these conditions, oxygen is, that means in these conditions, hemoglobin is readily binding with oxygen. And what are these conditions and where we are observing? These conditions we are observing in alveoli. What are these conditions? These are less temperature, means when compared with the tissues, at the alveoli temperature is little bit less. So that's why they are saying like this. Next one, partial pressure of oxygen is very high. At the alveoli, oxygen partial pressure is very high, and partial pressure of CO2 is very less. Next one is H plus ion concentration is less. H plus ion concentration is less means pH value is more. Yes or no? pH value and H plus ion concentration are inversely related, right? H plus ion concentration is less. That means pH value is more. And not only this, 2,3-diphosphoglycerate. Even though in many books they are giving diphosphoglycerate, it should not be diphospho, 2,3-bis. 2,3-bis phosphoglycerate. Technically the word bis is better and correct. when compared with the word di, but still many books they are using diphosphoglycerate. Let us use the same thing, but it is bis. Okay, but because at two different locations phosphate is attached, that's why bis. When you will use di, to the same carbon, suppose second carbon, to the same carbon two phosphates are back to back attached, then you should use di. Already these names, where how exactly we are using, already these names I have explained when we studied about the nucleotide structures. I hope you can remember that. Now let us see this is also more here. So these are the conditions, so this is less here, this is less here. So these are the conditions, that are seen at the alveoli, and because of these conditions we are observing left side shift when compared with standard graph, and this is conclusion is this one. That means at the alveoli there are some conditions, because of these conditions at the lesser partial pressure itself the oxygen is able to bind to with hemoglobin, so that at the alveoli we are observing that hemoglobin has more affinity to bind with oxygen, so that it is readily binding with oxygen and forming oxyhemoglobin. So these are the factors that are required for oxyhemoglobin formation. These are the factors required for oxyhemoglobin formation. Okay, so here 2,3-dipga is lesser here. Now exactly these are opposite at the tissues, but before directly writing this, first we will understand the meaning of this particular graph. Here, what is the meaning of right side shift? Because of the right side shift, what actually we are observing is, to get the same 50% saturation, much higher oxygen partial pressure is required. What is the meaning of this? More partial pressure of oxygen is required means what? Hemoglobin has less affinity for oxygen binding. Less affinity means what? Hemoglobin can easily dissociate. So here it is dissociation of oxygen from hemoglobin. So there are some conditions we are observing at the tissues, and they are responsible for this, so that oxygen is readily dissociated, separated from hemoglobin, so that it can easily enter into the tissues. But here at the alveoli we are observing some conditions so that oxygen can readily bind with hemoglobin. These are important for examination, and these are exactly opposite to this one. Here what we are saying, temperature is high, partial pressure of oxygen is low, partial pressure of CO2 is high, and H plus ion concentration is high, that means pH value is less, and this 2,3 diphosphoglycerate or bisphosphoglycerate, whatever it is, this value is also high in RBC. These are some things, and these are the fact exactly opposite to this. Remember any one, don't try to remember both of them, and again you will make mixture of all these things in exam. Don't do that. Remember for one, so exactly opposite for the other one. Is it clear? So in this way, this is what the significance of P50 values. Lesser P50 value, what is the meaning of this? Oxygen binding with hemoglobin is more. That means hemoglobin has more affinity to bind with oxygen. Higher P50 value, what is the meaning of this? Hemoglobin has a less affinity. That means dissociation chances are more. In this way, P50 value will give a better indication of what type of conditions are present. This is how we are using these experimental values for our understanding purpose. So that's why you need to understand which side it is shifting. So these are the P50 value significance, and at the alveoli what are the conditions that are responsible, at the tissues what are the conditions that are responsible. All the information we have covered, and with this transport of oxygen is completed. Clear? So now we will slowly move. This one already we have completed, and now we will go with comparison with other conditions. What are these conditions? Now observe carefully. This was not directly mentioned in NCERT, but they can ask in examination. So now you need to understand this concept also. Now let us see this. So this is the standard graph. This is the PO2, partial pressure of oxygen. This is the percentage saturation of hemoglobin with oxygen. Let us see. Now we will talk about the standard graph. Standard graph for hemoglobin A1. What is hemoglobin A1? It is having 2 alpha chains and 2 beta chains. the one that is having 2 alpha and 2 beta chains. That is what the standard graph is. Now, when compared with this adult hemoglobin, generally they may give HbA, still it is the same one because 97% is this one only. So that's why HbA, adult type. This is what the graph is. Now, when compared with this, where you will find the fetal hemoglobin? Fetal hemoglobin, HbF, fetal hemoglobin, this has alpha 2, gamma 2. Please observe this. Oxygen binding affinity is not same for both of this. It is not same for both of this. Not same means both the graphs will not overlap. So, for fetal hemoglobin, on which side you will get? Is it on left side or right side? Please remember, if you are writing on left side means this will have more affinity. It can readily bind with hemoglobin. If you are drawing on the right side, it has a lesser affinity to bind with hemoglobin. Now think logically on which side you need to write, on the left side or on the right side. Now which one has more binding affinity? What is the concept behind this? Please remember, these are all interlinked concepts. You should never ignore these type of concepts. Though not directly mentioned in NCERT, there are huge chances that they can ask these questions directly in assertion and region. Then you cannot escape. So now let us understand the concept. And this concept, anyhow you know about erythroblastosis fetalis concept. It is not directly linked to that, but some basic information, let us understand this. Suppose this is the mother and this is the fetus. When we studied about erythroblastosis fetalis, we have very clearly discussed this concept, and there itself I told you several times in the regular lectures, and once again I am telling you, observe carefully. Between the mother and fetus, there is a barrier, it is called as a placenta. In short, I am drawing the rough diagrams, so please observe carefully. Yes or no? Next one. Now here, the mother blood is coming up to the placenta. Mother's blood is coming up to the placenta. And then there is a placenta that is separating. And after this, there is an umbilical cord that is connected to the fetus. What is the question here? Is the mother blood directly entering into the fetus? Is the mother blood directly entering into the fetus? No, it is not directly entering into the fetus. Then the question will arise. Since mother blood is not directly entering into the fetus, then how fetus will get its own oxygen? How fetus will get oxygen? Oxygen is required. Now here there is another question. That another question is, within the fetus also lungs are getting developed. Lungs are present here. Within the fetus, lungs are slowly getting developed. But the question is, are these lungs functional? Are these lungs that are present inside the fetus, are they functional? First thing is, these are non-functional. Non-functional, please remember this. Even though lungs are getting developed, they are not functional. When they will be functional? After the delivery of the baby only they will start functioning. Until then they are not functional. That means these lungs are not useful. These lungs are not useful in breathing process. So how fetus will get oxygen? So fetus should get oxygen from the mother only. Then the problem is, mother blood is not directly coming here. Then how fetus will get this oxygen? Now this concept is interlinked. Observe carefully. This is the mother blood. Mother's blood is having HbA, that means alpha 2 beta 2 type of hemoglobin. Whereas fetal blood is having HbF, it is having alpha 2 gamma 2 type of hemoglobin. Now please understand, now fetus should forcefully take oxygen from the mother. Yes or no? Understand the logic. Fetus should forcefully take oxygen from the mother. That means when compared with mother hemoglobin, adult hemoglobin, fetal hemoglobin should have more binding affinity. Then only it can take oxygen from the mother. Yes or no? Understood the logic? That means between these two, who is having more affinity? Fetal hemoglobin is having more affinity. Now we understood the answer. If fetal hemoglobin has more affinity, on which side you need to write? On the left side you need to write it. On the left side of this you need to write. This is the left side. That's why this is having more affinity. So that's why now let us see the graph. And this is the graph for HbA, adult hemoglobin. On the left side of this they have given fetal hemoglobin. Why it is on the left side? You should know the answer. All these are interrelated concepts. If you don't know the concept, you will make the mistake. Even though you forget, now in the examination if they ask based on the concept, you need to develop the graph. That's what the concept we need. Majority of the students and teachers, they are thinking that biology is a by heart based subject. It is not like this. 70 to 80% is concept based only. Just 10 to 20% is memory based. If you are trying to memorize and remember, or remember means if you are trying to by heart the concept, definitely you will make the mistake. So don't try to by heart the concept. So in this way, these two are giving sigmoidal shape only, but fetal hemoglobin on left side and adult hemoglobin on the right side to this. They are present like this. Okay. Here we are talking another one. What is this one? Myoglobin. Why we are talking about this? This is also important. Please remember, this is what the interlinking is. We are talking about myoglobin. For the myoglobin, you are getting hyperbolic. These two are sigmoidal shapes, but myoglobin is giving hyperbolic. First question is, why you are talking about myoglobin here? Very important. That is the reason why regarding the myoglobin, I told you under biomolecules tertiary structure of protein. Hemoglobin, these two are hemoglobin. 4 subunits are present. 4 subunits are present. Quaternary structure you are observing here. In the biomolecules we studied. But myoglobin is having tertiary as the highest because it is a single protein. In the hemoglobin, 4 proteins are there. 2 alpha, here it is 2 beta, but here it is 2 gamma. Whatever it is, 4 chains are there. But in the case of myoglobin, only single protein is there. Single protein is there. Understood or not? So here it is all of a sudden it is getting completely saturated. But here it is sigmoidal. In NCRT, under the exercise, they have asked a question. Why hemoglobin is showing sigmoidal? Why hemoglobin is not showing hyperbolic like myoglobin? Because here 4 chains are there. Entire hemoglobin is not attached at a time. First one oxygen molecule will attach. Attachment of one molecule will enhance the affinity further. So for the attachment of first one, it is taking more time. For the attachment of second one, further lesser time. For the third one, further lesser time. For the fourth one, still lesser time. So in this way, there is a concept called as positive cooperativity. Attachment of one oxygen molecule will help in attachment of other oxygen molecule very quickly. So that's why it is slowly rising. So this is what you are observing. Understood or not? So that's why positive cooperativity concept we are observing here. That's why oxygen hemoglobin dissociation curve is sigmoidal curve. But myoglobin is giving hyperbolic because it is just a single protein. But here 4 proteins are seen. All these are logical questions. Read NCRT, even exercise questions also you need to solve. Clear? This is what the concept is. I hope now you understood how exactly we need to use this. So now I have given this particular answer. Why oxygen dissociation curve is sigmoidal for hemoglobin and hyperbolic for myoglobin. Hemoglobin has 4 chains and a positive cooperativity is observed. But myoglobin has single. So all of a sudden it is showing saturation. Clear? Next one. What are the factors that are affecting dissociation curve, and what are the conditions at alveoli and what are the conditions at tissues? Just now we have discussed all these things. Clear? Next one. Calculation of oxygen transport. Just now I told you, 100 ml of blood is having 12 to 16 grams of hemoglobin, and 1 gram of hemoglobin is carrying 1.39 ml of oxygen. That means approximately 100 ml of blood is carrying. So here it is carrying 20 ml oxygen. So this is for oxygenated blood we are carrying. So that's why 100 ml blood is carrying 20 ml oxygen. Though it is carrying 20 ml, only 5 ml is delivered to the tissues. So remaining 15 ml is carried by deoxygenated blood. So the word carrying or carries and delivers, these two are completely different. Carrying means it is with it. Delivers means giving to the tissues. So observe carefully. Don't make any mistake. Okay. Calculation part also very clearly I have explained everything. Next one, with that oxygen transport concept is completed. Regarding the CO2 transport, if you are observing, what is the first one? Partial pressure. Here also partial pressures we are observing, but if you are observing in the case of oxygen 159, 104, 95, 40. Likewise, we are observing the huge differences for huge partial pressure differences for oxygen. But for CO2, we are not observing that much of partial pressure difference. 45, 40, 45, 40. Like this only we are observing. That means partial pressure difference is very less. Please remember, if the partial pressure difference is more, diffusion will be more. But in the case of CO2, partial pressure difference is very less. Now you may have the doubt. Then how more CO2 will be diffused? Here that partial pressure difference is less, but solubility is much higher. Second factor we studied, no, solubility. When compared with oxygen, carbon dioxide is having 20 to 25 times higher solubility. Now it will play important role. If the solubility is more, even for a less partial pressure difference also, more CO2 will be transported. This is how it will be compensated. So this logic also we need to interlink. Anyhow, membrane thickness is same and it is very thin, so diffusion can occur very easily. So these are the 3 points that you need to link up here. And here 7% is directly dissolved in the plasma and 70% is transported as bicarbonate and 20 to 25% is transported as carbaminohemoglobin. So in the case of carbaminohemoglobin, this hemoglobin in this form, CO2 is not attached to Fe+2. In the oxyhemoglobin, CO2 is attached to Fe+2. But here it is not attached to Fe+2. It is attached to amino group of these protein chains. Amino group of these protein chains. That's why it is forming carbaminohemoglobin. Understood or not? And here also Fe is present in +2 form. Whether it is oxyhemoglobin or carbaminohemoglobin, whatever it is, Fe is always present as Fe+2 only. Very important for examination. Remember this. Next one, what is this particular concept? Roughly I will try to explain you this. I don't want to go into each and every point. In simple, I will try to explain you this. Suppose these are the tissues. These are the tissues and tissues are producing CO2 here, and this CO2 is entering into the blood vessels. This CO2 is entering into the blood vessel. Yes or no? CO2 from this is entering into the blood vessel. Now initially it is present in the plasma. When it initially present in the plasma, 7% is directly dissolved in the plasma and remaining approximately 93% is entering into RBC. Suppose this is the RBC. Okay, it is entering into RBC. Now this CO2, out of this CO2 what happens? 70% CO2, what it will happen? CO2 combines with water and in presence of an enzyme. What is the enzyme? In biomolecules also we studied that name. Carbonic anhydrase. In presence of this, it is converted into H2CO3 carbonic acid. And this carbonic acid is further hydrolyzed into H++ HCO3 minus. HCO3 minus. And all these things are happening inside the RBC. Now this HCO3 minus will come into blood plasma. And since one negative ion is coming into the blood plasma, to compensate this, one chloride ion is entering into the cell. So charge is compensated on both the sides. And this is called as Hamburger's phenomena, and it is also called as chloride shift. Chloride shift. And now this HCO3 minus that has entered into the plasma, it will combine with Na plus Rk plus, and finally it is forming NaHCO3, sodium bicarbonate. If it is combined with potassium, it will form potassium bicarbonate. So in this bicarbonate form, 70% it is going to the lungs. So 7% dissolved form, it is going to the lungs. Clear? Then what about the remaining? So approximately 20 to 25%. Now this CO2 is attached with hemoglobin and it will form carbaminohemoglobin. In this form it is going to that lungs. Understood or not? So in this way CO2 is going to the lungs. At the lungs what happens? This is directly dissolved form, it will go into the lungs directly, into the alveoli, no problem. But this one, reverse reactions will occur. Reverse reactions will occur. Once again they will bring back this CO2 that will come into the plasma and that will go into the alveoli. So whatever the reactions that are occurring at the tissues, exactly opposite will occur at the alveoli. So in this way CO2 will be released into the alveoli. In this way CO2 from the tissues is going to the alveoli through the blood in various forms, and these are the different things. This is in simple. Clear? So this is what actually we are talking here. So this is what the reaction that they have mentioned in the NCRT. And ultimately what they are saying, 100 ml of deoxygenated blood will deliver how much? 4 ml of CO2. Even the partial pressure difference is very less. In deoxygenated blood 45 mm mercury, in the alveoli 40 mm mercury. Just a 5 mm mercury difference, but still 100 ml is able to deliver 5 ml of CO2. How it is possible? Because of more solubility. Understood or not? This is what, anyhow you don't need it, but still I am saying, in the deoxygenated blood 52, in the deoxygenated blood, so actually this 4 ml, right? In the deoxygenated blood, every 100 ml blood, 100 ml deoxygenated blood carries 52 ml CO2. This is for deoxygenated blood. And from this what happens? 4 ml into lungs means alveoli is going. So 100 ml oxygenated blood, 100 ml oxygenated blood carries 48 ml CO2. This is what the original calculation is. Understood or not? For the oxygenated blood, what we said, with respect to oxygen, 100 ml oxygenated blood carries 20 ml oxygen, and it is delivering 5 ml oxygen to tissues. So deoxygenated blood will carry 15 ml oxygen. This is what you are saying. But for the CO2, this information was not there in NCRT. Simply remember this, that is enough. 100 ml deoxygenated blood carries 52 ml CO2, and it delivers 4 ml of CO2 to the alveoli for release, and hence oxygenated blood, it is carrying 48 ml of CO2. This is what actually the original calculation. In NCRT, simply they are talking about how much it is delivered. So you just focus on that one, that is enough. Okay, this is what they are saying. Next one, the Bohr effect concept we are talking is regarding oxygen release at the tissues. So what are the factors that are required for oxygen release at the tissues? So that you are talking here. And Haldane effect is, we are talking with respect to how exactly CO2 is released at the lungs. So once it is transported to the lungs, so how exactly it is released into the lungs. That concept we are studying regarding Haldane effect. Next one is Hamburger's phenomenon is the chloride shift. HCO3 minus from RBC is coming into the plasma, then CO2 is going, Cl minus is going into the RBC. Exactly reverse is happening at the alveoli, right? So this interchange of this HCO3 minus and Cl minus in opposite directions is called as chloride shift or Hamburger's phenomena. Clear? With this, the transport of oxygen and CO2 are completed. Now let us come to the last part, that is called as regulation of respiration. In the NCRT, they have not mentioned dorsal respiratory group, ventral respiratory group. In simple, in NCRT they said that in the medulla region, this is the medulla region, and above this is the pons viroli. In the medulla region, there is a particular portion called as respiratory rhythm center. Respiratory rhythm center. In NCRT directly they said, but actually here it is having dorsal respiratory group and ventral respiratory group. Dorsal respiratory group will control the normal breathing, and the ventral respiratory group will involve in forced breathing. This is what actually internal detail. But in NCRT simply they said that there is a respiratory rhythm center that will control the rhythm of the breathing. That's it. Simple point. And next one, within the pons there is a portion that is called as a pneumotaxic center. And what actually the pneumotaxic center will do? Very, very important. Pneumotaxic center will help in inspiration cutoff. What is the meaning of inspiration cutoff? Try to understand this. Suppose I am doing the normal breathing, you just understand this. Like this you can understand. In the normal breathing, I am doing normal inspiration. But in the normal expiration, now this respiratory rhythm center is controlling. If there are some conditions where inspiration should not occur for this much of time. Now let us see this. Suppose I am doing the running. When I am running for longer duration, what happens? Can I do the inspiration for the normal duration? No. The inspiration should cut off very quickly. So I am doing, yes or no? So once I am doing inspiration, there it should stop. Immediately I should go for expiration. So in this way, some signal should come to stop the inspiration after a particular time. Yes or no? So in this way, pneumotaxic center is the major one that is involving in inspiration off switch. inspiration off, inspiration should stop there, so that immediately we should go for expiration. In this way, what happens? Breathing rate increases. So whenever we are doing this running and all, what happens? CO2 levels will increase, H plus ion concentration will increase. We need to remove them. For the removal of this, now quick respiration should occur, faster breathing should occur. So in that case, this pneumotaxic center is very important. This is what the significance of inspiratory off switch, inspiratory cutoff signal. So this is what they are saying. But though not mentioned in NCERT, there is one more thing that is called as apneustic center. This is actually acting opposite to pneumotaxic center. So these are some things, but as per NCERT, they are talking about respiratory rhythm center present in the medulla region, and then pneumotaxic center is present within the pons region. These are the two things that they have mentioned. Along with this, within this medulla portion, there is one more thing that is called as a chemosensitive area. What this chemosensitive area will do? Please understand this. So in the case of heart, I am drawing one rough diagram. So observe carefully. Suppose this is the heart. So now let us see this one. So this is the left ventricle. Yes or no? This is the left ventricle. From the left ventricle, systemic arch or aorta is coming out. So within this aorta, and also there is a carotid artery that is going to the head. Carotid artery that is going to the head. Now in these two, actually these are carrying what? Oxygenated blood. And within these, there are some portions. They are called as some carotid bodies. So there are some portions. Within these portions, some receptors are present. And what these receptors will do? They will measure H plus ion concentration, and then they will measure the partial pressure of CO2. Please remember, once again I am telling you, actually these vessels are carrying oxygenated blood. In the oxygenated blood, these are present, but their concentration or partial pressure should be less. In the oxygenated blood, H plus ion concentration and partial pressure of CO2 should be lesser. But if these receptors are present, now they will constantly measure these values. If suppose we are doing some exercise, or I am doing some running or some activities, then what happens? The levels of H plus and CO2 will slowly increase, even in the oxygenated blood also. Then what happens? This information, this is the chemosensitive, these are some chemical molecules, right? Now they are sensed, and that information will come to chemosensitive area that is present once again in the medulla region only. Respiratory rhythm center is present in medulla, chemosensitive area is also present in the medulla. And once these values are high, and that information will be given to respiratory rhythm center, so once again this breathing rate will be altered according to conditions. Yes or no? So in this way, H plus ion concentration and partial pressure of CO2, for these only the receptors are present. But technically, if you see, oxygen, for the measurement of oxygen, receptors are not present, because even though oxygen levels are very high, there is no problem to our body. But oxygen levels are less. Oxygen levels are less means indirectly CO2 levels will be very high. H plus ion concentration will be high. Anyhow, sense for the sensing of that, receptors are there. So again our body will adjust the breathing rate or respiratory rate. So that's why the role of oxygen in the regulation is very less. This is what the another sentence they have mentioned in NCERT. With this, regulation part is also completed. Now we are going with the disorders. Some names they have given. Simply we need to understand this. Asthma is the difficulty in breathing that is causing wheezing due to inflammation of bronchi and bronchioles. In the case of asthma, the inflammation of bronchi and bronchioles are taking place. That's why the patient will face the difficulty in breathing. This is what. Next one, emphysema. In the case of emphysema, the alveoli will be damaged, and the major cause for this one is the smoking. When the alveoli are damaged, exchange of gases will be affected. So that's why it is a serious issue. And then if you are talking about the interlinking concept in the biotechnology applications, in class 12th, so there they are talking about a protein that is called as alpha-1 antitrypsin. This alpha-1 antitrypsin is used to treat emphysema. This sentence was given in biotechnology applications. So this emphysema we are studying here. So interlink these two concepts. Clear? Next one. So this is what about normal conditions and the emphysema conditions. There are some examples. Next one is regarding occupational respiratory disorders. So in some occupations, generally the individuals are exposed to some dust like things. dust and some glossy fiber like things in different industries like the grinding, stone breaking, and then asbestos, silica industries. In those industries, so they are exposed to some irritants, and directly they cannot cause any problem, but over a period of time now they are getting accumulated and our immune system is not able to remove those things. So in those cases, those particular portions will be damaged and they may lead to some fibrosis. That means it is the proliferation of the fibrous tissue. So ultimately the exchange of gases will be affected, the lung will be damaged, the individual will suffer with breathing issues. So generally, so it is suggested that such type of individuals who are working in those industries, they should wear the protective mask so that to some extent they can protect themselves from these dangerous things. That's it. These are some standard examples that they have given in the NCERT. With this particular chapter is completed. And before concluding, one more suggestion is, please don't try to by heart the concepts. Try to understand the concepts. Suppose they are talking about some partial pressure values. Anyhow, those values you need to by heart, but the remaining concept you need to understand. Clear? And once again I am saying that, so these video lectures are available both on Live Prep app and also on our YouTube channel, but the test series can be given only through the app. Thank you. So with this it is completed. So in the next lecture we can meet with another chapter. Thank you.